Right.
Let's see if that works.
Hi, everybody. This is Dr. Chris San Miguel, and I am going to introduce you to this year's systemic topic, which is malaria. Malaria is a life-threatening infectious disease spread to humans by some mosquitoes. Not all, but some. And it is caused by a parasite that's carried in the mosquito and is found mostly in tropical countries. This disease is both preventable and curable. And there are 5 different Plasmodium species that cause malaria in humans. 2 of which are the focus of most of the research and preventive care because they are the most prevalent it's called a vector, something that transmits the disease. So an important fact is that malaria has killed more people in history, over the course of history, than any other disease. So this is showing you how the Plasmodium parasite uses both humans and mosquitoes as part of its life cycle. So the mosquito will bite a human, and in doing so, it will kind of spit into the human. I know this is gross. And in doing so, it delivers sporozoites from the Plasmodium parasite. Okay, so this is a very early stage of the parasite. The parasite then moves into human red blood cells for further development. It goes through more stages. And when another mosquito comes along and bites that same human, when they draw up the blood, they're also drawing up parasites. So the sporozoites that the second mosquito sucks up with the blood transform into gametes, transform into zygotes, ultimately to oocytes, okay, which have more sporozoites inside of them. Okay, so then the process continues. When that second mosquito then lands on a new person and bites them and spits into them prior to blood, they will transfer this Plasmodium parasite to the new host, to the new human. All right. So after the female mosquito has had a blood meal, has sucked the blood of a human, she will lay her eggs either in or very close to standing water. Mosquito eggs and larvae can't survive moving water, so the fact that it's standing water is important. The eggs can survive dry conditions for a few months when they have the right moisture conditions, wetness. The eggs will hatch and the mosquito larvae will enter the water, and they're called wrigglers. The larvae will live in the water and feed in the water, and the life cycle will then progress to the pupae or tumbler stage. The pupae live in water as well, but do not feed. And after 2 to 7 days as a pupae, an adult mosquito emerges from the water surface and flies away. Depending on the conditions, this life cycle that I've just described can take 2 weeks, but can also be shortened. That length of time can be shortened to just a few days to as long as a month. Again, it depends on the environmental conditions. All right. Symptoms of malaria can be mild or severe, or anywhere in between. Mild symptoms are headaches, fever, chills. So similar to the flu, but severe symptoms include fatigue, confusion, seizures, vomiting, difficulty breathing, It can cause death. It is a fatal disease. It can be a fatal disease. But in areas where it's endemic, meaning it's always around, people will often get sick multiple times a year, depending on their climate. So if they have a couple of rainy seasons during the year, they will often get sick during each rainy season. This becoming sick requires medical treatment, which can be very expensive and will take people away from work. So here are families that are often already in poor situations, and then they're losing their income and having very large expenses. So it becomes a huge financial burden for families. Okay. To lower the risk of getting malaria, the advice is to avoid mosquito bites. This has often focused on nighttime, on sleeping conditions, because historically mosquitoes have been active at night. So mosquito nets are highly advised, and they can come with mosquito repellent built into them, with insecticides in them. There are also, of course, spray mosquito repellents. We've all used DEET or something like that. Off is our brand here, or one of them. The use of coils and vaporizers. So again, putting an insecticide into the air, either through coils. I don't know if any of you has seen the little coils where you burn the end and it's a slow burn, but it's an insecticide. And then, of course, to wear protective clothing and use wind screens, because a lot of areas of the world, they don't use window screens. And vector control. So again, a vector is an organism that transmits a disease from one place to another, or one host to another. So mosquito control is highly important in malaria control. So two main interventions for vector control have been the use of insecticide-treated nets and indoor residual spraying. So insecticides are used and sprayed. You'll see in pictures later on that people will often have netting hung around on the walls, and that netting will be sprayed with an insecticide so that it stays in the house and it stays on the walls. And insecticide treated bed nets, as I mentioned, cover the sleeper. Okay. There are anti-malarial medicines available. These should be administered to vulnerable populations at periods of greatest risk, such as during rainy seasons. The issues here are often getting to the people who need it during the time periods that are needed. Infrastructure is often poor in a lot of places where malaria is endemic. So there aren't good roads, there isn't good public transportation. People have to drive or figure out how to get to medical facilities. In order to do that, they may have to take a day or two off from work. So it becomes an issue. And I've listed there the different types of chemotherapy, and chemotherapy just means treatment with a chemical. But again, a lot of people who need these anti-malarial medicines are not receiving them. So for decades, the incidence of malaria and deaths from malaria were dropping. Okay, so you can see this line where on the graph where it's going down, 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 down. It hit a low, malarial deaths hit a low in 2015, and since then have been on the rise again. And there are two main reasons for this. The first is that mosquitoes adapt and evolve quickly. So phenotype is the expression of genes, right? So an organism can have a gene for blue eyes and a gene for brown eyes, but if they have blue eyes, then their phenotype is blue. Though they do have the genes for both, right? So mosquitoes, the phenotypes have been selected against those that are active at night because they can't get their blood meal because of these measures that people are taking, such as insecticide-treated nets. And so that phenotype is disappearing, and the phenotype that has evolved is that there are the ones that are active more during the day. And we'll get into that more. The second reason is climate change. So the area where malaria carrying mosquitoes are found has been expanding. And in fact, for the first time in 20 years, in the summer of 2023, malaria was reported in the United States. Okay. And in 2020, there were 627,000 malaria deaths worldwide. Okay. So the genetic and phenotypic makeup of mosquitoes evolves quickly in response to environmental change, environmental conditions. And mosquitoes have developed resistance to all currently used insecticides and are more active during the day and biting during the day than they used to be. So the phenotype of nighttime biters has lessened because they can't find people to bite because of the mosquito, the bed netting. So the ones that are active during the day have the advantage, so they proliferate. And then genetically, they develop resistance to these insecticides because you kill the ones that are susceptible, and then the population of the ones that are not expands. Okay, the parasite itself, the Plasmodium parasite, is also increasingly resistant to the drugs that were once highly effective against it. Okay. In 2022, there were 249 million cases of malaria, 94% of which occurred in Africa. And there were an estimated 608,000 malaria deaths globally. 80% of those deaths were children under 5. So malaria is one of those diseases that impacts the young and the old and the people in between. It tends not to be a fatal disease for them. And you can see from the map, red is obviously a high number of malaria deaths, and green is low. So in October of 2021, the World Health Organization, abbreviated WHO, recommended broad use of the first malaria vaccine among children living in regions with high Plasmodium falciparum malaria transmission. It was shown to significantly reduce malaria and deadly malaria in young children. A second malaria vaccine was recommended by WHO in 2023. It's too early to tell the impact of these vaccines on malaria in the regions where they're being used. And there are other ones that are in the pipeline. The problem is that often these medicines are expensive. Again, they're hard to get to. If you need boosters, the parents are having to take multiple days off from work, which sometimes isn't financially feasible. So yeah, so again, we'll see what happens with these vaccines. They haven't been in use for very long. Another problem that is recently appearing, is that malaria was always thought to be a rural disease. and that it didn't really impact people in cities, partly because of different conditions, environmental conditions that weren't as conducive to mosquitoes. And the fact that if you're in an apartment, you're more likely to have window screens, things like that. But a new mosquito species, Anopheles stephensis, is appearing in urban areas. It's adapted to urban environments, and it can survive long dry seasons. So this is a new vector for malaria that is being found in urban settings. This new vector, stephensi, I guess, breeds in standing water. And as you can see, here's a cistern full of water that is just standing water. And it thrives in congested cities where there's unreliable piped water systems, okay, resulting in water storage. There's also poor trash collection. So when there are things like bowls and plates and bottle caps that can hold small amounts of water, the mosquitoes will use it as a place to lay their eggs. Now, when you consider the population of urban areas within Africa, particularly, you are increasing the number of people vulnerable to mosquito-borne infections by more than 100 million. Okay, so that's a significant increase in the number of people who could potentially contract malaria. Researchers are developing new approaches, some using a new generation of tools to biologically and genetically modify the mosquitoes to block the Plasmodium parasite. These efforts are extremely costly and often face regulatory hurdles, particularly things with new techniques. So, for example, you all know that AI is causing a stir because the government hasn't caught up with the technology to regulate it properly. The same sort of thing can happen with new treatments. The government just doesn't have a review process, so it doesn't get reviewed in a timely fashion. And remember, these organisms are adapting. So if you take 10 years to develop a vaccine, chances are your vaccine, by the time it comes to market, may not be effective anymore. All right. So there are some innovations being tested. One is called spatial repellents. So if you can see, that gentleman has a piece of plastic that he's pinning to the wall. These are filled with a low dose of a chemical that confuses mosquitoes and prevents them from biting. And they have been proven effective. One of the issues with them is that they have to be replaced every 12 months. So it can end up being, if the cost of these things is too high, it will be prohibitively high for people with very low incomes. These nets, by the way, that are hanging on the wall, the draperies, they're likely treated with insecticides as well, because this is the indoor residual spraying technique. Another new innovation is called an attractive target sugar bait, or an ATSB. It's about the size of a sheet of paper, and you can see those little black pockets within that sheet. Those are filled with a liquid that's very sweet and is laced with insecticide. It's a new kind of insecticide because, remember, these mosquitoes have developed resistance to all currently used insecticides. And all the insecticides currently are from the same class of chemical. Okay, so that you have to change the way that the chemicals are, how the chemical functions. So the mosquito, in theory, will smell the sugar in this liquid, will take a drink from it, and then it will die because there's an insecticide in it. However, you need multiple of these. They're hung on the outside of the houses, but they have to be kept away, obviously, from children, from livestock. So they're put higher up on the walls. You need one at least on every side of your house, if not more, depending on the size of the house. And they have to be replaced every 6 months. So again, you're talking about potentially having requiring a large investment from the family in order to use this method. Spraying, you see this person is spraying a field with insecticides. So for a new insecticide to work, obviously it has to be safe for humans. It has to come in solid form so that it can be distributed easily and not dissolve in water, not dissipate in the water or get diluted. So, and as I mentioned, it has to kill the mosquitoes in a different way than the existing class of insecticides. Because the mosquitoes have developed a resistance not just to the different chemicals, but to the way that the chemicals are killing them. And since it's a single class of insecticides, pyrethroids, they're all killing in the same, using the same essential mechanism. Okay. All right. There are also new bed nets that are being introduced. The one that this woman is holding up is called Interceptor G2LN. So it is a long-lasting insecticidal net that combines a new insecticide with the pyrethroid old insecticide. So now this net has two different ones. And it lasts, as I mentioned, the bed nets generally need to be replaced once a year. These apparently can last 3 years based on the company's testing, just to let you know. All right. There are genetic approaches that are trying to alter the vector's ability to transmit the parasite to humans. So this is showing a transgenic is the mutated mosquito. Wild type is the regular mosquito, and you can see in the bottom, okay, that the way that they're doing it, all of the offspring are going to be mutated to not be able to carry the to not be able to transmit the parasite. They can still carry it. So it showed promise, but they haven't been able to move it forward since 2015. There's nothing else been published by this group on this method of modification. More recently, a research team engineered a mosquito that actually blocks the malaria parasite, so it's not even carrying the parasite. And they're using something called gene drive inheritance as opposed to regular inheritance. And by modifying both genes on the chromosome, all of the offspring will end up with the modification. Okay, so the idea is that you're increasing the spread of this gene so quickly that within a few months, basically, you're going to be able to prevent the Plasmodium parasite from entering any of the mosquitoes which would be lovely. This is one of their test subjects. You see it's covered in green dust. And they intentionally did that so they could identify, they released them, and then as mosquitoes were biting people, they collected them to see which ones they ended up with. So part of the problem with this, even though it sounds like a great idea, people in governments are often wary of genetic modification. And as I mentioned, they don't have regulations for it, so they don't know what to do with it. So instead of doing anything, they do nothing. So one country's health minister said that his government was focused on basic needs like water and electricity, and that genetically modified mosquitoes were a luxury his government couldn't spend time or energy on. So the real world use of these mosquitoes, even though they develop them and they can produce them and release them, this is on hold indefinitely. All right. So mosquito-borne diseases, there are a lot of them. Okay. So another example is dengue fever. And there's a group of researchers that have found that a common bacteria, Wolbachia, which occurs naturally in about 50% of mosquitoes, of insect species, including mosquitoes, can fight viruses. Okay, so dengue fever is an example of a virus, a viral disease. Now, that's not the case for malaria, but if a female mosquito that has Wolbachia has eggs, she will pass Wolbachia to all of her offspring. Okay. Wolbachia, certain strains of Wolbachia have been shown to block the transmission of viruses. So as I mentioned, dengue is one, Zika virus is another. And once the bacterium is introduced to a population of mosquitoes, breeding those mosquitoes is fast because the females pass the organism down to all of her eggs. And so it's also very inexpensive relatively. Okay. These mosquitoes can then be released into the wild, and they will impact the wild population, right? So they will breed, and then all of the Wolbachia mosquitoes will have Wolbachia offspring. Okay. Because these are not genetically engineered organisms, and because Wolbachia is a naturally occurring bacterium, the release of Wolbachia mosquitoes has very little limitations put on it. There are few restrictions. Okay, so one of the things that's not known at this point is, are there bacterial candidates that target the Plasmodium parasite? So could you do something similar with, say, another Wolbachia strain, or with a different bacterium that may impact the Plasmodium parasite? Some researchers believe that only large-scale environmental management and housing changes will make a difference in malaria occurrence. So things like draining breeding sites, and you can see this house is essentially in water. It won't be all of the year, I'm sure. It's just during the rainy season, but all of this water will then be standing and will be a great place for mosquitoes to breed. Larvicides are also effective, so you'd have to have large spread larvicide spraying programs to kill the larvae in the water. And larvicides don't cause resistance or haven't yet caused resistance. And then, of course, moving homes. So that's a big endeavor, and who's going to pay for it, right? Part of better housing that's needed. You see here, there are two houses in this picture. The one on the left has been plastered. The brick walls have been plastered so that all of the holes and cracks have been sealed up and mosquitoes can't get in from between the bricks. The doors and windows have been framed out so that the doors and windows sit tightly and are sealed when they're closed. Windows that open are fitted with window screens to keep mosquitoes from coming in and out while still allowing for ventilation. Metal roofs keep the mosquitoes out better than the thatch. Apparently the mosquitoes can just find routes through the thatch. The other advantage of a metal roof is that it can last 20 years as opposed to a thatch roof, which it has to be replaced every 1 to 3 years. And then sealed rather than open eaves. So eaves are where the walls and the roof meet. And if you go back in this talk to the frame where I was first talking about innovations, and there's the gentleman hanging up that piece of plastic that's impregnated with a chemical. You can see that he is living in a house that has open eaves. So you can see the sunlight coming in from between the wall and the ceiling, the roof, and mosquitoes can just fly right in through that. All right. So I hope this has given you some ideas of what
All right. So I hope that gives everybody a good idea of the issues of malaria, the extent of malaria, and where our students started with their innovations. So we are going to start with I've gotten a request that we start with the Rutgers, New Brunswick team. And I'm trying to find, their name is Are You Stemming? So do we have the Are You Stemming team?
So he's on. We have Matthew, and I'm going to share his presentation for him. So
okay, so let me. All right, let me go here. Let me go to Kiana. All right, so Kiana, you can share your screen, and Matthew, whenever you're ready, you can start your presentation.
Thank you so much. Can you hear me?
Yes.
I'm sorry, I just want to preface this with the fact that I'm at a research conference right now, so I have a little bit of noise around me, and I need to turn off my camera just so I can use my data a little bit.
Okay, yes.
So welcome everybody. Thank you for having us. We are Project Ripple, coming from the Are You Stemming team at Rutgers, New Brunswick. My name is Matthew Parchment. This project was developed with Anaya Elliott, as well as Chelsea Abade and Santiago Mascara. And we would like to continue to show you what malaria really is and how we wanted to tackle that problem. Next slide, please. So as you already saw in the video, over 229 million cases of malaria globally and 600,000 plus deaths annually. Mosquito breeding is the problem specifically that we decided that we could tackle directly, specifically with stagnant water, as it's a perfect breeding ground for mosquitoes. And current solutions that they have right now around net sprays, medicine, and focus treatments aren't necessarily for prevention, but what happens after the fact. So we thought that our best bet and the best bet that we can give to people around the world was to prevent it rather than treat it directly. Please continue. So as we learned, female mosquitoes seek out stagnant water or still water to lay their eggs, and these environments provide a necessary condition for the development of mosquito larvae. This is because with the stagnant water being stable, it allows the eggs to be in the safe environment for which they were laid already, which may be in a tree log, a rotted tree log, a tire, or whatever it is. It allows it to be stationary. Next, it protects because the water is not moving, it's less oxygenated. Therefore, you're not as likely to have predators in that water, and it saves the mosquito larvae again from having to compete with other organisms in the environment. Third, you have a lot of bacteria in the environment typically, which not only helps provide nutrients for the larvae, but it prevents other organisms from being able to thrive in the area. This also is one of the reasons why we have things like malaria being bred with the mosquitoes, because they tend to lay their eggs in such infested areas. Please continue. Next slide, please. So the solution that we have is a water agitator. So the Ripple specifically is a derivative of current water agitators that are on the market. The Aqua Vortex is a solar-powered, low-cost water agitator that prevents mosquito larvae from maturing by adding a ripple in the water, allowing it to move, disrupting where they are, allowing the water to oxygenate and just change the general environment to make it suboptimal for mosquito larvae. Please continue. So water agitators come in many different sizes. You have permanent ones, like these bigger agitators on the right, and then you have temporary ones that you add based on season and depending on where you are. Sometimes they can be very expensive. Maybe that we might have a small farmer in another country, a third world country, that doesn't necessarily get large rain seasons or has unpredictable rain seasons. It wouldn't be cost effective for him to add this huge agitator system into his area where he doesn't know how much rain he'll have like from time to time. So having something that is portable, detachable, and customizable would be very effective. We go to the next slide, please. So this is where we developed the Ripple. You see a side view right here on the top. We have a little solar dome. This solar dome is essentially what we use to capture photo electrons, what powers the device specifically, in a similar manner to how a calculator would work. On the bottom, we have on the lower view, it's sort of like a speaker system or a system in which we can induce rippling by storing the energy, passing it through a capacitor, and releasing it through some type of piezoelectric. And then the above view, you can actually see what it looks like. You see we have a clip system. So this is our innovation, which is the clip system. We would be able to attach multiple of these ripples to each other dependent on the location and dependent on the amount of water that we need to affect. So in some cases, in some villages, you may have small puddles that accumulate, and you can use one or two ripples to handle the entire area of water. In other cases, you might have abnormal amounts of flooding, and you may need more. So you can, instead of having to buy multiple and put them all around, you can attach multiple ripples together, and through the mechanism of sonic resonance, we would be able to create bigger waves and bigger ripples to attach to larger bodies of water.
Time.
Okay, so I basically did go through all our functions. I would like to say thank you on behalf of Project Ripple. Are there any questions to the functions and possible future innovations that we have continuing with this?
That was great. Can you tell me, can you go back one slide? So what is this supposed to represent here?
So
Why
future
did you
innovation. I'm sorry, continue.
Why did you include these particular pictures?
It was just to present an understanding of nature, some things that are biodegradable, that are nutritious, and an understanding of how you can build it by yourself at home. So an idea is for a nutrient-infused outer shell that allows for passive diffusion into the plant. So the original material that we designed this our device out of would be a hemp polymer uh, based material. But if you want to, you could also use hydrogels, like in the fabrication of the material, adding like adding a nutrient base that would naturally diffuse into the water source that you want to apply it to. So not only are you keeping mosquitoes away, but you can help feed plant life as your as your product slowly degrades into the water itself. And then you would just have to remove the whatever wiring casing that we would have placed in the machine as it like slowly degrades over time from the water. And then along with this, we have alternating frequencies. So alternating frequencies has this picture up top, which shows like how you can have it. The goal is that maybe mosquitoes could eventually evolve or like develop some sort of resistance to whatever device we have. Maybe we can change the frequencies, change to add like more of choppy water, maybe something that would be a little bit more disruptive to like the nervous system or of these mosquitoes as they're developing. And then we have also a build at home kit. Maybe we've seen multiple kits where you're able to print your own 3D printer at home. It sounds like hilarious. How are you able to print your own 3D printer? Sometimes you can go to like other locations and you're able to print certain parts of whatever machinery you want. Maybe we can develop our kit such as that, where we just send the wiring over and we send you the solutions that you need to print the hydrogels in commercial 3D printers and whatnot. And that's kind of why we use these pictures, is to show nutrients, a home environment with the speaker in which you could have made it yourself. You have like complete control over that. And then also the biodegradable aspect of it, where it's reusable in this last picture, showing that it's a symbiosis between technology and nature.
I love the idea of the nutrients because by encouraging the plant life, then you're oxygenating the water, which makes it less of an environment that the mosquitoes enjoy. That's a great addition.
Exactly. Thank you so much.
Tom, do you have any questions? Oh, you're muted.
No, that was very, I just unmuted. I think I unmuted myself. Yeah, that was very innovative. I have no questions.
Okay.
Nicely done.
Very nicely done. Does anyone else in the audience have questions? No? Okay, then we shall move on. Thank you so much. Everybody give them a round of applause. The next team up is Bad Mosquitoes. And they are from NJIT. And just to let you know, I've been getting all kinds of requests to go first or second or next or whatever. There is an order that we have, unbeknownst to you. Somebody can't go first. So we're just going to follow, unless you have a reason, like one of your members has a test later or whatever, unless you have a reason like that, we're just going to, after Bad Mosquitoes, we're just going to follow the order that we had. And actually, Bad Mosquitoes was supposed to go second anyway. All right, so who's here for Bad Mosquitoes?
We have 3 people. We have myself, which is Maddie, Andrea, and then we also have Haley, and Andrea is getting these slides up right now to present.
Andrea Gomez?
Yes.
Okay.
Yes, I'm right here.
All right, you should be able to share now.
All right, so good morning, everyone. This is Team Bad Mosquitoes, which consists of me, Maddie Stevens, Haley, and Andrea. And we just wanted to, I guess, give a brief introduction on malaria and that it is a life-threatening disease, as we all know, caused by the Plasmodium parasites, which are transmitted to humans through the bites of infected female mosquitoes. It remains a major global health concern, especially in tropical and subtropical regions where mosquitoes thrive due to warmer temperatures and stagnant water. We also know that mosquitoes are found worldwide, but they are the most abundant in warm and humid climates. They lay their eggs in or near the standing water, such as ponds, marshes, rice fields, and even artificial containers like buckets or discarded tires. The larvae develop in water before emerging as adults, making it easier to access or making their access to water crucial for their life cycle. So, mosquitoes are drawn to specific environmental cues when searching for a host. They are the most active during dawn and dusk when temperatures and humidity levels are favorable. In an ecosystem, they are attracted to warm-blooded animals that emit body heat and carbon dioxide, and mosquitoes can detect carbon dioxide from exhaled breath up to 50 meters away, making it a key signal for locating hosts.
Sorry, guys.
Okay.
To tackle mosquito populations at their source, we propose a new mosquito trap designed to take advantage of mosquito behavior and their natural attractants. The trap consists of a black plastic cone wrapped with mosquito netting that has very small holes large enough for mosquitoes to enter but difficult for them to escape. Inside the cone, a carbon dioxide chamber slowly releases carbon dioxide through a small hole covered in wax. The wax controls the release, gradually softening due to a heating pad. The heating pad is powered by a solar panel, ensuring continuous operation without an external power source. At the base of the cone, there is a bowl of soapy water where mosquitoes eventually fall and drown. The soap reduces surface tension, making it impossible for them to escape. These traps are standing about 5'3" to 5'11" feet tall, will be placed around bodies of water that have been previously monitored and identified as active breeding areas.
Using Autodesk Inventor, we were able to create a CAD model of our design based on the previous sketches. The CAD model uses functions such as revolve to replicate our desired design. The first image on the left depicts the entire structure with the mesh net. The second image in the middle shows the trap without the mesh netting, and the final picture on the right is a model of the carbon dioxide canister that would be in the center of our design.
Who goes next?
So, utilizing prior knowledge of mosquitoes and their ecological habits will help us to use this to our advantage. So, basically, attracting them to the carbon dioxide chambers preys on their instinctive preferences, thus allowing the trap to exploit this and strategically decrease mosquito populations. And essentially, less mosquitoes means less malaria. The incorporation of solar panels also allows to create a freely generated power system and also making the contraption available to less funded areas while increasing the prevention of malaria. Thank you.
Excellent job. That's really interesting. All right, so I have two questions for you. The first one is, you have a solar panel to heat the wax, but a lot of the places where malaria occurs and is endemic are hot places. So what temperature were you thinking, and what happens if the external temperature is higher than that?
Because of the places that malaria is usually found in very hot, I would believe that it's not hot enough to completely melt wax to the extent that it's completely like dripping through. I believe that with like the heating pad, it would just like I believe like maybe a temperature of like um about 97 degrees to fully just soften the wax. because of the fact that it's not directly on top of the plastic cone, it's just behind it, and it would just slowly, gradually release heat. It would not just be always giving heat. It would just be like a small, continuous going like spikes of heat.
All right. And what if you're in an area where it's 105 degrees outside? What happens to your system?
Then I believe the solar panel would just not be needed, but it would still work in the sense of it would still be a gradual release of carbon dioxide.
Okay. And then my other question is why the height of 5'3"?
Because of the fact that we're trying to replicate a human body and like trying to mimic a host, we're just kind of looking at average human heights to make the traps, to just
Okay.
kind of like seclude them in a specific area.
Okay. Excellent. Tom, any questions?
No, very
Okay.
innovative again.
Anybody else have questions? Come on, guys, you should be listening to one another and this should be like your time to question.
I have a quick question.
Sure.
So you mentioned that the CO2 need to be replaced. How often would that be? And as well, if it's near bodies of water, what happens when it floats?
So the carbon dioxide I've read from different people who have used them for like, for example, aquariums that does like the gradual release. It can last from 8 to 10 months before it has to be replaced. I would say that the only thing that needs to be replaced at least consistently would be the soapy water, which is at the bottom of the cone. And also, these are not going to be directly on top of the water. They're just going to be next to the water, bodies of water surrounding it on land. And since these are just like big sticks that are just going to be kind of like stuck on the ground, um, I don't see a problem with them starting to float or anything.
Thank you.
Okay. Anybody else? All right, that was great. And I'm sorry, in future, can everybody or turn on their cameras so that we can actually see you guys as well as your slides? It just makes it more personal. So going next is Team PCCC, which is Brigitte and Judith, and I'm not certain who the last one is.
Hi. So let me just share this. Can I share this slide?
Hang on, let me get you as a,
Thank you.
there we go. Go ahead. You should be able to.
Hi, everyone. Good morning. My name is Amira,
My name is Bridget.
My name is Judith.
and we're a team from PCCC. We have heard so many wonderful presentations already. For us, it may not feel like that much time has passed, but around the world, these minutes could have been valuable to someone. Every 2 minutes, another life is lost to malaria. Right now, as we're speaking, another child, another mother, and another father loses to this disease that is entirely preventable. We have the knowledge and the technology and the power. The time to act is now, and yet malaria continues to claim another 600,000 lives every year. But what if we could stop it at the source? So the area that we researched that has a very high malaria percentage is Nigeria. About 27% of the global burden accounts in Nigeria. A lot of this is due to the heavy rainfall and the high temperatures. Because of the heavy rainfall, there's a lot of stagnant water, which is the ideal habitat for the Anopheles mosquitoes, which are the primary malaria carriers.
Malaria is spread through the bite of infected mosquitoes, and without intervention, the cycle continues. The key to stopping malaria isn't just treating those who get sick, it's preventing the mosquitoes from reproducing in the first place. Anopheles mosquitoes lay their eggs in stagnant water, and these eggs hatch into larvae before becoming disease-carrying adults. By targeting larvae at the source, in buckets, water storage containers, and natural breeding pits found in homes and communities, we can drastically reduce the mosquito population and in turn, malaria transmission. We will be using BTI spores, a natural occurring bacterium that produces proteins that are toxic to the mosquito that are toxic to mosquito larvae when ingested, killing them before they mature into disease-spreading adults. But we don't stop here. Our formula also includes LS, an effective and environmental-safe microbial larvicide used in controlling mosquito larvae. These stay viable for long periods of time. With this innovative approach, we're not just reducing mosquitoes, we're disrupting the malaria cycle at its core. This is exactly what our product does, larvae capsules, and we're here to show you how.
To fight malaria in an eco-friendly way, we created a special capsule that kills mosquito larvae. The capsule contains 2 natural bacteria, BTI that works fast, killing larvae within the first 2 or 4 hours. Lasts longer, for example, the LS continues to kill larvae for several weeks. The capsule is made of biodegradable materials like alginate and chitosan, making it safe for the environment, and we also use calcium chloride to make it strong and stable. Our capsule works in 2 steps. First, BTI attacks the larva's stomach, breaking the cells and making it stop until it dies. Then LS infects the remaining larvae, damaging their internal tissues and weakening their muscles, so they can move or grow into adult mosquitoes. This way, the capsule kills larvae quickly and also prevents new mosquitoes from growing. Our solution is safe, so it's safe for humans and also is for the environment. It does not harm the environment at all.
To make a real impact, we plan to distribute these capsules in Nigeria, where malaria is a big problem. We will focus on urban and rural areas. We will distribute these capsules from June to October when mosquitoes are most active. People can easily use our capsules in any stagnant water. Some of the advantages of our capsule is its dual action effectiveness, it's eco-friendly and safe, it is biodegradable, it's sustainable and cost-effective, it's targeted approach, and it's very affordable. Our mission is to ensure that anyone anywhere can use this capsule as a powerful tool in the fight against malaria, empowering communities to take control of their health. Thank you.
Thank you.
Thank you.
That's excellent. Really, really great idea. I love the fact that you're using naturally occurring organisms. And do you know for a fact that the BT bacteria and the LS bacterium attack the larvae?
Yes, there has been studies shown that these BTI spores, the new innovation is adding these 2 bacteria together, because the BTI does work, but it's only effective for a certain amount of time. So the LS would increase the amount of time of effectiveness to kill these larvae.
Really, really interesting. And it's fabulous that it's safe for
Yes, yeah.
all different creatures. Like, you know, you have a fish pond, you don't necessarily want to put something in there that might kill them, or if you have animals and it's your livelihood, they're your livestock, you don't want to risk hurting them
Yes.
with a thing. Would you have any instructions for the farmers or ranchers or whoever was using this as to when to apply them and at what dose to apply them?
Yes, there is one slide that shows the product. Okay, usually it's in the 7th.
Oh, right here.
Yes, perfect. So we can see our idea is to present like in pearls spheres. That is basically is like for 10 liters, one of those spheres. It could last between 2 to 3 weeks. It could be refrigerated and last 6 months. So that is the idea.
Okay.
So yeah, the packaging would include kind of the instructions of storage. How would you use this? Where to apply it to?
I love your packaging too. That's fabulous.
Thank you.
Tom, questions?
This sounds very economical and easily managed. Do you have any idea what the cost is?
$2.
How much?
$2.
$2.
$2.
Yes, we wanted to use
For
10,
40 capsules.
yes, 40 capsules.
That's amazing.
$2.
Yes. So
Like
40
for-
capsules would the total would be $2 to make 40
Yes.
of these spheres.
For a family is one envelope for family
Mhm.
is the idea for this kind of period of time.
And I assume there's no reason why this is limited to Nigeria. It could work pretty much any place.
Yeah, it could work anywhere.
Any.
This is just where your testing area.
Yes.
Yes, definitely.
Wow.
Okay, anybody else? Questions?
I have a quick question.
Sure.
So theoretically, these capsules, they would need to be applied to every stagnant water source in order to be effective. What happens, however, when you have water sources that are more difficult, like not as easily accessible in regions where distribution of basic materials become an issue, and also after it rains? Because in the beginning of the video, it was stated that the life cycle in some cases only needs a few days to complete. And like, is it realistic to be able to apply these to like every puddle or stagnant water source?
So for example, our idea was that it would be not 100% effective, but mostly effective in most water sources. So it would be applied to water where there's already like mostly large water sources where there's already like a high chance for malaria. And in areas where malaria is already prevalent, they would be given more thought and they would already be given more capsules. Obviously it can't be in every single water, but we're trying for like the most success possible.
But that's the reason why we are using the BTI and LS, because the BTI is quick and is not too effective in the UV rays, but the LS is more resistant and also is resistant in stagnant water. So that's the reason why we are using the two ingredients.
Okay, thank you. And I would just like to comment that I do like how you guys sought to incorporate both the fast and slow release spores to increase the efficiency of the timeline. I think that was a really good approach.
Thank
you.
Yeah, you did a great job. Thank you.
Thank you.
All right. Next up is supposed to be the HCCC blue team. Are they here? And if not, is the green team here?
Yes, the green team is here.
Okay, so we'll go with the HCCC green team.
Okay.
Who needs to
Hi.
share?
We will give us 1 sec for you.
Yeah, it's just
Okay. Oh, and you're all together.
Yeah.
Hi, Dr. Lee.
oh, this is share. Yeah, the camera is already on.
Spin like this. Can you see us? Is everything?
We can see you.
Yes.
But I'm not getting a request for you to share. And what's
Yeah.
the name? What is the name of where you're at? Like, what is your name? Oh, it's just in the corner. I see it. Okay. Let me make you co-host, and maybe that will help. All right. So now you're co-host. So you should be able to share now.
Okay, click and
There we
click
go. No.
That's the share on camera. It's not the share here. I don't have anything yet.
So you don't have a share button?
I saw it before it was here.
Not on our.
Oh yeah.
That's not. Press it. Okay.
Can you see us? Can you see
We
our
can see
screen?
you, but we can't see your presentation.
Okay. Give me 1 sec. Would it be all right? Thank you. No.
It might be in more.
No, I don't have anything to share our screen.
We click share, but it doesn't share.
Okay, so Amaris Rivera is the name that I have as co-host.
Yeah.
So you
Yeah.
should be able to share.
You can try.
Wait, you want to close this and do the share again?
Sorry about that. Give us 1 second.
First, the green team is from where again?
Hudson
They're from-
County College. Sorry about that.
Well, let's not, how about just look at the screen here then, because we have a backup.
It may be that you're projecting it and that may be the issue.
Oh, we use 2 different computers.
Oh, okay.
Would it be all right if I shared it with you?
With me?
Yeah, that way we
Yeah,
can.
go ahead. You can
Thank-
send it to me and I can try and pull it up.
Thank you. Let's see. Did you get it on your end?
Not yet.
Okay.
No, I still don't.
It's in the chat.
No, I sent it to you privately.
Oh, it's in the chat?
Yeah.
Okay, hold on.
Sorry, I just thought that'd be quicker.
Yeah, probably is. Okay, let me share. Okay.
Perfect.
How can I put this back on our screen?
Okay, so let's go.
Thank you.
There we go.
Yeah, make it big
All
so
right,
you-
go ahead.
Okay. So thank you for your time and letting us set that part up. So let's just start then. We are the HTCC Green Team and we're here. to
Okay, wait, before you continue, it's better if you're closer because we're seeing I know it's helpful for you, but we can't really hear you so well when you're that far away.
Yeah, can stay louder.
Is
okay?
this better?
Yeah, that's fine.
Yeah, come closer.
Okay. So I'm gonna come real close, okay? So thank you for coming today, everyone, to the Systemic Challenge. Now we will move on to our agenda overview, and we're going to go through all of these slides for you one by one. So let's go to the next one and tell you the big idea. Advance. Thank you. So here is the big idea. We're looking to have natural vector control. As you can see, this is one of our professors who has helped out our team as well, and that is the Passaic Valley Sewerage Commission, who came to help build our nesting boxes. Now, why nesting boxes? Birds eat mosquitoes. It's a part of their diet. So the idea is to build little to no cost, very time efficient, and anyone can do nesting boxes. And we're going to show you how we do that.
You may be wondering which bird we chose and why, and the answer to that will be the barn swallow. The reason we picked this bird is because they can eat up to 850 mosquitoes a day, while only being about 11 inches in height. Another reason we picked them is because this is one of the more prominent species of the swallow, so they can be found on any continent around the world, so it'll be easier to implement that. And additionally, they're not very picky about where they live, so they'll live anywhere that's like pre-hollowed or just an empty-ish area. So that on top of their small size, they're more susceptible and more likely to live in the bird houses that we could create for them. So it wouldn't be too hard to get these birds to be put around mosquito-filled areas.
So for our building process, we use materials such as wooden pallets, nails or screws, and other tools that could be used to build a nesting box. For this building process, we had the assistance of Chris Brook from Passaic Valley Sewage Commission, and he helped us put the wooden pallets together with nails. So in the bird box, there are holes drilled to the top in order to hang the bird box with zip ties or another durable material. And there's a hole in the bottom of the box for drainage.
as for affordability, the per box, it would cost us $2. And if we were thinking about using other tools and materials, it would cost a bit less, as it is also effective in building nesting boxes. As for paint, it is unnecessary, but it does increase the longevity of the box, and it is an optional extra cost. It could be a fun activity for children as well. And as to recap the entire presentation, nesting boxes will attract more barren swallows to an area to control mosquito populations, and we believe that it will naturally reduce the case of malaria because of how many mosquitoes they eat per day. And for the building process, it will take approximately 1 minute and 30 seconds, and this may vary due to the tools and materials you use and if paint is included. And for any wood or tools used, it does make it an inexpensive process and something everybody can do.
So we do intend to have future work with this. Educational outreach is a big part of it, especially with Passaic Valley Sewerage Commission. Our team wants to make sure that people do know about this, and that leads into our environmental welfare. Now, this is because if you're more hands-on with your environment, you'll care more about your environment. So we hope that people will be more attentive. And we have experience working with grants and writing proposals with other nonprofit organizations, and that's something else that we'd like to do with this. So, thank you. Thank you for listening.
I apologize, I wasn't advancing the slides. No one was telling me to, and I didn't realize until this slide that you guys were on a different one.
No, it's okay, because we have someone here advancing our slides for us. So it wasn't for you.
But no one else got to see them, so.
But
apologies.
please, yeah, show everyone. Yeah.
So I'm just curious. This is a DIY like flat pack thing that would be sent out in pieces with all the components, and the individual using it would be the one to put it together. Is that correct?
So it can be, that is a future input that we would like, but the main idea is that anyone can do this with any material, anywhere, because it's such a simple process, and because swallows don't necessarily have a certain preference for where they live. So it can be anywhere in the world. If you happen to stumble across a wood pallet that isn't needed, you can break it apart and use it with anything that you have.
Okay, that's fabulous. And so you design plans basically.
Yeah.
That could be distributed so that you're attracting this particular bird to the nesting box.
Yes, and specifically, can I see the box, Andre? Thank you.
Can you ask them to show the slides again?
Yeah. Can we show the slides again to everyone just while we go through this?
Yeah, so which slide do you want me to be on?
Let's start. Yeah, let's start at the beginning one, and then we'll just slowly go through it again.
Okay. Well, okay. You only had 5 minutes, so you can't give your whole presentation again.
No, we won't do the whole entire thing.
You're at 344.
May I have your question as well?
Just that it's specific to the bird. The box design is specific to the bird.
Yes. So they have a 1 inch and 1/4 diameter, so this is perfect for them. As well as when they go in here, they can do whatever they want, because after a while, the drainage will help for any other birds. It'll get rid of anything else that's inside, and some birds will also still use what may be left inside. So it is specifically for the swallows, since swallows are everywhere.
Okay.
And any other questions?
Not for me, Tom.
So did you work with an ornithologist, and this ornithologist told you that these birds are endemic throughout the world? That's all.
Oh, okay. Sorry, I didn't want to cut you off. So we did our own research about barn swallows, and they are abundant around the world. Yes. We didn't particularly work with
fascinating birds. Yeah.
Yeah.
You know them well. Yes. Nicely done.
Thank you. Any other questions?
All right. Nope. Well, so any other questions? Everybody out there? Anybody want a question? No? Yeah?
Okay.
All right. Well, thank you, PCCC. No, HCCC Green Team. Sorry.
Thank you.
All right.
Good job.
Great. This is up next. NJIT EOP search, are they available? No, then we can substitute. Oh no, then we can skip that and go to William Patterson STEM stars. Are you guys here? Jason, Cecilia?
Yes, we're here.
Okay.
Hello. We're just gonna have one of our teammates share their screen.
Which
Just give us
one?
a second.
So I can.
That will be me.
Jason. Okay.
Jason.
All right, you should be able to share now. All right,
Okay,
let me see it.
perfect. So first, we want to start off by saying welcome to everybody. We hope you guys are having a great morning, and we've been loving all of the presentations. You guys are all very talented and smart individuals. And our names are all Jason Peralta, Jelani Rogers, Janine Sanchez, Cecilia Vasquez. We come from William Patterson University and all have diverse backgrounds in biology, computer science, and environmental science. The reason for our innovation is because, as we know, malaria remains one of the deadliest diseases, especially in Africa, South Asia, and etc. Despite medical advances, there are three major challenges: late detection, poor tracking, and lack of prevention efforts. Providing communities with real-time alerts, system tracking, and prevention strategies, we could save millions of lives. What is Malaria 360? Well, we created an app that is AI-powered and can detect, predict, and help communities fight malaria because we believe prevention is more important than cure.
Malaria 360 is designed to support individuals and health organizations with two key functions. The first one is easy data reporting. So users can report symptoms, cases, and mosquito breeding sites through our mobile app, website, or SMS for those without smartphones. The mobile app and website offer simple ways to log symptoms, locations, and other details. Second, instantly be able to process reports to detect outbreaks patterns. So this data can be used for the interactive map and risk zones, which will show high-risk areas in real time, helping local communities.
So in regards to the market opportunity, health organizations and NGOs benefit greatly from Malaria 360, as early detection can save lives by enabling faster treatment and intervention. So local governments can use this technology to implement similar malaria control policies, ensuring that resources are allocated effectively. High-risk communities also stand to gain as real-time alerts and prevention strategies help reduce infections and improve public health outcomes. Potential partners such as the WHO, CDC, and Red Cross can support global malaria efforts by integrating Malaria 360 into creating initiatives, while national health ministries can leverage the data to enhance response strategies. With strong partnerships, Malaria 360 has the potential to revolutionize malaria prevention worldwide, making a lasting impact on global health.
Malaria is still a huge global health problem, especially in Africa, where 94% of the 263 million cases in 2023 were reported. Nearly 600,000 people died from it that year, and young kids under 5 are the most at risk. The fight against malaria is struggling because there's a massive 4.3 billion funding gap, making it harder to prevent and treat the disease.
Why does Malaria 360 matter? As Gen Z students, we've grown up in a digital-first world where smartphones, apps, and online platforms shape how we connect, learn, and share information. Creating Malaria 360 was a natural step for us, using the technology we know best to address a critical global health issue. Access to information is power, especially when it comes to public health, yet many communities still lack reliable, real-time updates on disease outbreaks like malaria. By developing a mobile and web-based platform, we're ensuring that essential health information reaches people everywhere. You may ask, why an app? Well, for one, accessibility, malaria prevention tools and resources available to anyone, anywhere. 2, real-time tracking, instant data collection and outbreak monitoring. And 3, community empowerment. It allows alerts and prevention tips to help people take action. Our generation understands the power of digital tools, and we believe technology should do more than entertain. It should save lives. With Malaria 360, we're proving that innovation and awareness go hand in hand, ensuring that everyone has the knowledge they need to protect their health. Now, some impact of Malaria 360. Some of the key features that we have are real-time tracking for early outbreak detection and faster response, offline and SMS access so remote communities can report cases, crowdsourced data, which will allow lower cost, freeing funds for treatments, and scalable partnerships, which will allow expansion to impact across malaria pandemic regions. Malaria 360 makes prevention faster, cheaper, and more accessible.
We believe in the slogan, see something, say something. That's why when creating this app, we wanted to make sure it wasn't something that just stayed within our country, but something globally. When a new disease comes up or a new pandemic, it usually goes beyond control because everybody doesn't know. But if we all stay on the same page, then we can truly change the face of medicine. With support from global health organizations like WHO, CDC, and Red Cross, Malaria 360 can revolutionize malaria prevention, protecting vulnerable populations and move the world. Thank you.
Anytime.
Thank you.
Great job. So I have I have one big question. So how is this used by a community that is, or an individual, because they might be so spread out that they don't really form that kind of community that we think of? How do they report something? Because, you know, you're in the middle of a place where there's no paved roads, there's no electricity, there's no cell phones, you know, so you said it could be used offline for reporting. What would that look like?
I would definitely say, I mean, being that we would have an app tracking this, and you're in an area or region that typically doesn't have, obviously, those type of accessibility, we would have it. We'd have to take it back to the basics. We're going on paper. We're having to get that information because at the end of the day, it still has to be reported in these areas of these typical or these types of outbreaks. So we'd still have to go into these areas and still gather that data and that information. So that way, again, even if you don't have accessibility to the app, you're still able to report it to this region and be able to at least alert and make others aware of it. I mean, at least that's ideally what we would like to do, and hopefully be able to create some type of accessibility to where we could create, and well, we could gather that data.
Okay. Tom, questions?
No.
All right. Any other questions?
Hi, yes, I have a couple questions.
Sure.
So my first question is, I may have missed it, but how do you, with your app, handle the potential misinformation that could be spread on an app that is able to update information from anybody?
So we would use AI-driven pattern analysis and cross-verification with official health records to ensure data accuracy.
Thank you. And my second question is, it's going to be a follow-up. So just real quick, it's publicly updated. So this would mean civilians or any community member can update this app if they have it.
Right.
So in the collection.
Oh, go ahead.
So
no,
in the
I was
past.
gonna add to it,
yeah.
but I'll let you finish your question.
No, go ahead. This might change my answer.
So the first being like, okay, if it's offline and we want to prevent like false reports or how do we report a case, right? So we would use like our health organizations also to like get into these areas so they could really help report or trigger these alerts, which will enable and have like faster response and targeted resource allocation, but also because the app was technically offline in some remote regions, it would help kind of support like SMS-based reporting. So say you don't have accessibility to an iPhone, but you have some type of phone that can report like data texting, you can still allow users to kind of send that case data without the internet access. So this would kind of still give you that real-time update even in those remote regions. I don't know if that really touched base, so you can follow up if you need to. That's okay.
No, absolutely. I just wanted to kind of like get more a better understanding on, because in the past, when the public is given like the opportunity to share information on something that could be true or potentially untrue, that could
Right.
lead to a lot of self-fulfilling prophecy or mass hysteria, which can be an even greater detriment to those communities and the communities surrounding them. So I was just wondering if there was a way to combat misinformation or even get another authentication of that information, because not all information is true. And frankly, in a third world country where it's very scary to live in an area that could be soon endemic or become an epidemic from malaria, it
Yeah.
could be very potentially dangerous to pass on false misinformation.
Yeah,
That's just what I was
absolutely.
wondering.
Yeah, no, absolutely. And I definitely agree with that. And I think that's why we need to use things like we have to definitely utilize our organizations, making sure that not just public information, I feel like the public could then report it to these well-known, renowned organizations to verify this data. We wouldn't just like, I'm at home and I just sent you this, or I'm just posting this and now, it seems like it's an area that has this and it's not true. Because I agree with you totally. Like, I mean, we went through a pandemic. There was a lot of information that we were getting, whether it was true or false, but I think that's why we had to rely on our literal World Health Organization for verified information. And if we have these organizations in these remote areas to verify this data, because we don't want to spread misinformation. We don't want hysteria to be breaking out in these areas and people are getting even more worried. Listen, when the world shut down in 2020, we didn't know, we thought we had 2 weeks off and we were going back to normal, and we are here 5 years later. So I definitely wouldn't want the app to kind of misspread wrong information. So that's why we want to cross-reference our organizations and utilize AI. AI is new, it's coming up, but we really want to kind of track that data to make sure it's correct as well. So I hope that answers your question a little bit.
I also would like to add that's where our diverse backgrounds would come in. We have someone in environmental science, biology, and I'm also pre-med with also computer science. It's a group teamwork effort where not only one mind is thinking, but we're all thinking and trying to see what is accurate and using our own knowledge to be able to say, okay, this information that's being updated on the app isn't true. What can we do about it? How can we help them and calm them down so that the situation doesn't go overboard?
Thank you so much. I love the answer. That was really good. Really good,
Thanks
guys.
for the question. I think that's a great follow-up question because I think that's something to really consider in the day and age of technology is definitely misinformation and what we're typically consuming, and especially in those remote regions, like we want to make sure that we're providing correct information for sure.
Anybody else?
Have you identified a local agency within New Jersey that can help you with this? Get this thing up and running and distribute it and promote it and things like that?
That would be great. Honestly, I haven't identified actually, we could probably work with our public health. It could probably start with our public health organizations within our community, within William Patterson.
Yes,
And then as far as like,
public
yeah.
health is one of my minors. So when we came up with this project, I always mentioned we had to reach the public health officials. What can we do to not only have this information being spread in our community, but obviously globally? And that is one of the number one things that public health officials do, which is spread this information, because we believe that power is through knowledge. And yeah.
Right, that would lend some credibility to what you're doing too.
Absolutely. And I think as far as like just the technical side of it, I mean, we have a great computer science department, so we could definitely grab students to work on this code and really build it out. As far as, if we're talking about efficiency and really wanting to start up, having students work on something like this that will lead to a global impact. Yeah, I could definitely see us talking about this, presenting this at different, events, conferences. That's how, you get money, you get grants. That's what you gotta do.
You're giving this nice thought.
And it's great that your app is would be applicable across diseases. Like this doesn't have to be just for malaria. It could be for any disease. Like if you were working here in New Jersey, if we were having, a West Nile outbreak, or we're having a bird flu outbreak right now, it could be applicable to that as well, which is great.
Absolutely.
So thank you all.
Thank you.
Up next, we have the PCCC Dream Team 2.0. Are you guys here?
Yes, we're here.
Okay. Okay. Who is going to be sharing their screen? Or is it all of you all in one place?
Yes,
it's all of you all in one place. Okay.
Yes.
So let me make you guys co-hosts, and that way you can share your screen. All right, you should be set to share.
Can you hear as well?
Yep, we can hear you as well.
All right, perfect.
Okay. Today we'll be battling malaria with nature's solution. My name is Kathleen.
My name is Venus Like the Planet. I'm Stephanie.
I'm Samuel Vasquez.
And I'm Christy.
Malaria remains one of the most urgent global health challenges. In 2023, there were an estimated 263 million cases and 597,000 deaths across 83 countries. The World Health Organization African region was hardest hit with 94% of cases and 95% deaths. Tragically, 76% of malaria deaths in the region were among children under 5. Nigeria accounts for 27% of global malaria incidences because of tropical climates, especially from August to October, which are peak transmission periods and encourage an increase of Anopheles mosquito breeding. The prevailing Plasmodium species in Africa is Plasmodium falciparum and Plasmodium vivax. But predominantly, Plasmodium falciparum poses greatest threat because it affects red blood cells rapidly and has the ability to stick to blood vessel walls, making it harder for human immune systems to fight it.
As we have already heard about the environment in Africa and Nigeria, it's a favorable contributor for mosquito breeding. We also see how it's hot, it's humid, and there is a lot of heavy rains, which leads to flooding and standing water, and makes it more easier to have more mosquitoes. Vector controls as of right now are insecticide-treated nets, indoor residual spraying, but this has not been enough, and the fight continues.
Okay, but how does the fight start? Let's start with the transmission of the malaria. First, an uninfected mosquito is going to bite an infected person. That person is going to transmit the plasmodium in the mosquito, and that's what we want to fight with a bacteria called Delphia surohatensis TC1. This bacteria is going to infect the mosquito and is going to secrete a compound called harmane. Harmane is part of the family of the beta-carboline alkaloids, and what it does is that it stops the development of the parasite oocysts. Therefore, there is no formation of sporocytes. Here we can see in the first graph about the Plasmodium falciparum. In the y-axis, we have the oocyst number in the midgut, and we have in the x-axis the bacteria, non-bacteria, sorry, Delphia and Pantoia. We can see that Delphia surohatensis can inhibit 73.3% of the number of oocysts in the midgut, basically showing the effectiveness of the bacteria.
Which means that we'll have less or almost less Plasmodium in it. Our delivery system is going to be Bio3C1. This is going to be an encapsulated dry bacteria. We'll be using cassava starch co-processed with Avicel pH 101, which is going to help with the solvent effectiveness of the capsule. We are going to use cassava starch because it's a crop that is already growing in these areas. And we also are going to use flower nectar powder so we attract mosquitoes to these specific places. Although they already are attracted to water, we want to attract more. We are going to also have sealed powder packages for larger amounts of waters. And how it's going to work is that we are going to drop 1 capsule of 500 grams in the water, and it's going to dissolve. It's going to attract the mosquitoes to breed there, lay their eggs, and the larvae is going to absorb this bacteria And it's a symbiotic bacteria. That means that it's already in the mosquitoes. So we are going to increase the population of it and inhibit it from the whole lifespan. So that means it's going to continue throughout the whole lifespan of the mosquito, inhibiting the growth of the plasmodium. So we cut the cycle from the beginning, and they'll continue to breed with it, the resistance.
So CC1 in our product is very effective because one, it doesn't kill the mosquito. It only targets the plasmodium within it. And plus our bacteria that we'll be using CC1 does not require genetic modification, which can be expensive. It's also safe with the products that we're using, such as the dried bacteria, which is dead, and the cassava starch. And this will help to reduce the number of cases of malaria that we see that is burdening Africa, and also decrease the deaths along the way. And the only con that we have came up with is that there would just have to be a constant reapplication of our product to ensure the decrease of cases. And studies and trials must be done to ensure the efficacy over time. And just a reminder, our product is safe. BioTree C1 will be effective to implement into the community since one, it is inexpensive and it doesn't change their lifestyle, the way of living. And we can collaborate with organizations such as the World Health Organization in order to distribute our product, not just in Nigeria, but across many communities. And over time, we can see the impact and how TC1, our product, decreases the malaria cases, leading us to a better and stronger future.
Thank you.
Thank you.
Hello.
Sorry, sorry, that was me being muted. All right. So how do you think the reapplication is going? The need for reapplication is going to impact the not only the efficacy. That's not quite what I mean. What I mean is how well do you think people are going to adhere to having to reapply it?
I think that because we are targeting the plasmodium within self, so we are going to we're trying like to stop the actual disease. We could, I mean, I think like it's going to need a lot of more studies to try the bacteria within other materials, right? To increase the longeviness of its effectiveness. This is really new. This was basically, the study was released in 2023. So it needs more study. And I would say like if for any case we release it now, someone buy it, right, and we start producing it, it's going to be a matter of consciousness on teaching the population about what the product is doing. So the constant reapplication will matter. So and they know what are they attacking. It would only not prevent. It will basically at some point stop the disease because the mosquitoes won't have the development of the parasite.
Okay, excellent. Tom, questions?
Have you given any thought as to the cost and distribution?
Yes, so for the cost, because of the cassava starch, it's like 30 cents per ounce. That's very cheap. It will be around $2 or $7 for 50 capsule of 500 grams, and we'll need just one capsule per 20 liters of water.
And the duration of that capsule is for how long? So how frequently do you have to
It's going to depend on
the amount of water, I suppose.
Yeah, the water. And it will.
Are you going to provide instructions to the people who are administering this so they
Yes.
can figure all this out?
Yes, definitely.
You're liable to mess it up, you know.
With instructions, and also if we partner with the World Health Organization, we could distribute pamphlets on how it's going to be used. If they want to have controlled waters or standing waters, that also could be targeted.
Excellent. Any other questions?
I just have a quick question.
Sure.
Isn't cassava known for releasing cyanide when it's not treated properly?
That's why we want to, it's going to be all dry and use just the starch. Just for the solubility of it and the release of it to make it less dangerous. Yeah.
Okay, thank you.
It's just a vehicle. It will be just a vehicle for the bacteria.
Anybody else? All right, thank you, Dream Team 2.0. Up
Thank
next,
you.
up next we have William Patterson's The Catalyst. Are you guys here?
Yes, we are. We will have one team member share the screen.
Okay, who's the one that I should make co-host so that they can share their screen?
Nathalie.
Okay. All right. Nathalie, you are now co-host. You should be able to share your screen.
Okay, thank you so much. Okay.
You can go into slideshow.
Okay, yeah.
There you go.
Morning. Oh, wait. Sorry, my bad. Hello, good morning. My name is Ashley. I worked with Nathalie and Rafatu. We have a gold nanoparticle infused cream to protect against malaria.
So we know that malaria is a mosquito-borne disease that's caused by Plasmodium species in female anopheles. They claim more than 400,000 lives globally each year. Among the known solutions, sole mosquito repellent products reduce mosquito bites, but they don't often eliminate them. Early detection is challenging mainly because of the very low initial parasite number and asymptomatic early stages. So, by introducing a cream composed of hybrid nanomaterials, we were able to integrate it into one simple application that's designed for mosquito repellency and immediate therapeutic action. So we don't want to completely eliminate the mosquito population only because we know that they also have a purpose in our ecosystem.
So in our concept, we have three main ingredients that we want to incorporate in our cream. And those are gold nanoparticles, silicon sporozoid antibodies, and essential oils. Gold nanoparticles because they have been proven to have therapeutic applications in drug delivery. They also can bind to cysteine-rich domains in plasmodium surface proteins, and they are very stable in high temperature. So we will be using it for protein detection and delivery, also for stability. When it comes to silicon sporozoid or CSP, those are very abundant in those in the protein or sporozoids. And so, like, by targeting those CSP, we can try to neutralize them before they enter the liver. That's why we're using those antibodies for early detection and neutralization. For the essential oils like lemon and eucalyptus, we're trying to use the cream at the same time as repellent. Like, while it's neutralizing those parasites, it can also repel the mosquito to avoid them biting the populations.
So how it works, so gold nanoparticles functionalize with CSP antibodies. These highly specific antibodies are incorporated with the gold nanoparticles. Gold nanoparticles are proven to be antimicrobial and antiparasitic, which allows immediate neutralization once entering the skin. from a mosquito bite. In addition to that, we would have a natural repellent added in the cream, which is lemon and eucalyptus oil. So it is the only plant-based repellent recommended by the CDC for repelling malaria-carrying mosquitoes due to its active compound, p-methane-3,8-diol, which hinders their ability to detect human skin. So these main ingredients will be incorporated into a single cream, and users will apply this cream as a regular mosquito repellent.
You're muted.
Oh, I was saying it's very advantageous because those will provide immediate neutralization and like will have some therapeutic effect at the bite location, preventing the parasite to enter the liver. This is also cost effective because the gold nanoparticles are not expensive, and all these ingredients, we need them in a low amount. So that can be available for the populations who need these and don't have really like the funds to pay for very costly materials. And those will be stable materials ideal for tropical climates like high temperature. And it's a user-friendly, like you can use it every day in daily routines.
The way our cream mostly is going to be used as an over-the-counter product, there will be two types of bottles, one specifically for adults, another one for children. Another way we can distribute the bottle is for travel size. It is important that people who travel outside a lot or go to like specifically for Africa or any countries that carry the mosquito malaria, that they have one just in case with them. Obviously, they can't have like such a big bottle of material with them like in their carry-on, so it will become a problem when they go to TSA, or at least they have something that they don't need to buy like at a different country to have it with them. The points of a product is to be commercially available and safe to use daily. Those who do not have the resources to effect will have this solution readily available. Our cream will combat malaria and right away. It not only detects early stages, is a therapeutic solution with easy application, and it will be a very important tool for the public health and in endemic regions. Go ahead. Thank you.
Excellent job.
Thank you.
Great job. I love the fact that you thought about travelers, that people who are going to the region would also need something, and this is an easy thing rather than having to take very toxic prophylactic medicines like chloroquine, which is not used anymore because it's totally ineffective. Though it used to be like the go-to. So that's great. Do you have any, is there any research? Did you find anything on the long-term application of gold nanoparticles to the skin?
Yes, many researchers have been using it right now for like dermatologists, like to create creams for more applications. So it's being widely used right now in many like researches.
Yeah.
Oh.
And on top of that, gold nanoparticles specifically are being used in cancer therapy because they have, like I mentioned, like antimicrobial and so many other medicinal therapeutic properties. That's why they're being used in like other like diseases for like cancer and illnesses.
Interesting. Very interesting. I didn't realize that.
Also, instead of like silver, who is proven to be toxic, gold is being used because that's not toxic. So we can use it in our body to target many like diseases.
Very interesting. Thank you. Tom, questions?
Have you given any thought to developing some sort of a promotional campaign to overcome the natural resistance that some people might have to putting this cream? I mean, if they think it's killing, I know it's not killing the mosquitoes, but there may be some concern on the part of people that there might be some toxin in there that's hidden, and you might have to deal with that before they will put this not only on themselves but especially on their children. And I know you gave nice thought to the fact that you have both the formula for adults as well as for children. How did you happen to come up with that? And how did you change the formula from the adults to the children?
So, I think it would have to do with like the level of concentration, due to obvious reasons. For example,
So you haven't consulted with anyone yet.
we spoke to some of our professors on this. Yeah, so the level of concentration would be how we could decipher like how much we can use for like certain age groups.
So you have given this some thought. Very good.
Great job. Anybody else? Questions? Going once. Anybody else have any questions? Okay, well, thank you all. And next up, we have the team Ad Astra from Montclair. Quentin, Ado, Jean, and Tony Ann. Are you guys here?
Hi, good morning. Or good
Hi.
afternoon.
Quentin, are you the one who's going to be sharing your screen?
Yes, I am.
Okay, so I will make you co-host. All right, you should be able to share now.
All right, give me one moment. All right. Can you see my screen?
Nope, we see you.
Oh. No worries.
We've seen you before.
Good to see you guys again. Let's
You
try
had a
the.
good question before too.
Oh, thank you so much. I appreciate that. It was genuine.
Okay,
are you
now we
seeing...
can see it. Yes.
All right. Perfect. I'm glad. Just give me 1 second. Set up my end. All right. You ready? Nearly every minute, a child under 5 dies from malaria. Now, imagine if we could predict these outbreaks before they happen, saving countless lives and saving countless materials that could be expended efficiently and properly before this parasitic disease spreads. Our machine learning solution will forecast these outbreaks, enabling preemptive intervention and efficient resource allocation to these endemic areas. Join us as we revolutionize malaria. My name is Quentin Ocampo.
My name is Aldo Cruz.
I'm Jean Exantis.
And I'm Tony and Morgan.
And this is our model in predicting malaria outbreaks with machine learning.
So how the model works. The first step of our model is analyzing climate data. So we extracted a bunch of data like temperature, precipitation, and water vapor for specific regions in order to help train the model and identify what's causing the outbreaks. And then once we have this climate data, we combine this with the historical malaria cases, and then we use a random forest classifier to train the model to learn from past data so that it can identify the relationship between climate patterns and malaria outbreaks. And then finally, this is output on a geospatial map to easily see high-risk areas and showcase where malaria outbreak is more likely to happen so that we're able to take preventative measures and allocate resources effectively.
Here we now move on to data integration, where we will be collecting essentially 3 types of data. First, we'll start off with health data, which will include historical malaria cases, lab results, mobile tools, potentially apps, SMS systems, drones even. Next, we would move on to climate data, which would have records of temperature,
rainfall, humidity levels, where we could potentially use satellite instruments like NASA's MODIS. And lastly, we would also have geographical data, which would include GIS layers, analysis of vegetation, and proximity to bodies of water to really see how malaria is transmitted. And now here is an insight into the programming aspect. Right here is 2 snippets of a Python program. On the left here is the predictive modeling, which would essentially predict malaria cases based on a given set of data. And on the right is more so the visual aspect, which would include a map that would basically highlight high-risk zones of malaria outbreaks.
So why is our model important? Current malaria maps use historical data which can be outdated or incomplete. They can struggle with data integration from multiple sources such as climate data, health data, and geographical data. Both of these problems can lead to the lack of real-time updates. Both of sorry. And this is in no way, shape, or form downplaying current malaria maps. You see, this is just to showcase that our AI model will be more advanced, capable of providing real-time updates, so that preventative measures can be sent out before the outbreak occurs. So what do we want to get out of this model? We want to make sure that every resource counts. You see, our AI model identifies and predicts high-risk areas. This allows us to allocate our resources more efficiently, resources such as medical supplies, bed nets, mosquito repellent, and most importantly, information. With information, the communities of these endemic areas can be aware, they can prepare, and they can help prevent the spread of malaria. A prime example, COVID-19. Once people became aware of how to stop the spread of COVID-19, they started wearing masks, social distancing, and essentially we saw a decline in COVID-19 cases. Alongside this, the predictions will allow the deployment of medical supplies and personnel in a timely manner. Not only will we allocate our resources efficiently, we will also provide aid at the proper time, maximizing the impact of our efforts, leading to overall more effective interventions and minimizing that outbreak severity.
And again, to kind of follow up on that point on intervention, this program is going to be accessible for local governments and healthcare companies. So it's easy to access, it's easy to interpret. It's going to be localized information so that they can get this information ahead of time. Because, you know, more time gives them more time for proactive interventions and adaptations to the outbreak that could occur. And addressing community health, a lot of different areas that are going to become endemic or have an epidemic are especially crafted, culturally sensitive, so that we want to make sure that each area has a solution that's made for them. Now, let's revolutionize malaria prevention globally. And just like the systemic challenge for us, we want to follow this quote: start where you are, use what you have, and do what you can. Thank you.
Great job. Great job. I think that's amazing. You said culturally sensitive and it would be tailored. How do you envision that happening?
Yes, absolutely. I can answer that question for you, Chris. So in a lot of these areas, they have different cultures, they have different religions, they have different, you know, community beliefs. And tailoring our messages, tailoring our help to them, tailoring to these beliefs, customs, and languages ensures better community engagement overall for all of them, and it increases the likelihood for behavior to change.
Good answer. Tom, do you have questions?
No, so this is web-based, right?
I'm sorry?
This is going to be a website that you're creating for this to disseminate this information or make it available to other agencies and other users? Am I correct?
So currently it's a program, it's a machine learning model. It can be developed in either an app-based or a web-based platform, but currently right now we developed a prediction model, so it's a program and can be used in either format.
Okay. So you just have to work on the delivery method.
Absolutely. And to answer that second half of your question regarding who will we disperse it to, who will get our program or our developed website or app, that's going to be global health nonprofit agencies or just public health organizations such as WHO, World Health Organization, CDC, And to talk about nonprofits like Bill and Melinda Gates Foundation, who work heavily in malaria eradication programs and innovative solutions. But primarily, our goal is to make sure that our program is able to make these predictions ahead of time so that the local governments and the local healthcare companies in these soon to be endemic areas or predicted to be can get this information soon so that they can prepare for the upcoming epidemic.
So this is an information kind of gathering and prediction program that would be available to communities, health organizations, that sort of thing.
To be more specific, we wouldn't say communities, we would say to the local governments and healthcare companies, just because we believe that the best way to spread correct information and to take the right measures, and each government might have their own solution, or more so, they might have their own situation. So if we disperse this information to the local governments and healthcare companies, they might be able to tailor their own solution or tailor their own approach to the incoming spread of malaria. And we believe that informing them is important.
Okay. So like Red Cross would do something with it.
Yeah, that's a great example.
All right. Excellent. Anybody else have questions?
Yes, I do actually.
Go ahead.
So thank you for presenting. I love that you guys also had data machine learning. Machine learning. Okay, great. And thank you guys for sharing. So I guess my question is kind of just going back to like what my original, Chris had actually asked me this question originally with disparity in regions, right? So if you're in a rural area, how are you guys going to go about getting this information, getting this data to better source it? And I guess get it pushed out correctly for not necessarily your communities, but to your organizations. How would you kind of go about that with regions who don't have water, electricity, or accessibility to a phone? How would you necessarily go about that?
I would say that it would be mostly from like existing data, and we could also potentially conduct our own research. This would probably fall under the category of health data that I mentioned in my slide, where basically we would analyze trends within historical malaria cases or see laboratory results and potentially develop our own mobile tools to reach other communities and audiences that might struggle with malaria regularly.
Thank you for that. Oh, go ahead.
Oh, sorry. And about how will we reach them? Because it's understandable that in some of these third world countries, finding technology to reach them can be scarce or very difficult. But we will be targeting the local governments, and these local governments usually would have their own set of technology. However, again, like Jean mentioned, that we're going to look for mobile tools to go to these communities directly, possibly provide their local government with some sort of technology to receive this information.
Definitely. And I just have one more additional question. Like, how would you, I guess, measure the success of your app in terms of its impact with malaria prevention and awareness?
Yeah, that's a great question. So measuring the success for us, well, that's going to be over a long-term aspect, since this plan, this product that we have, this proposal is going to be a prediction. And since there are many factors that can influence a prediction, our goals with the new technological developments in the recent decade and in the future generation, that we're going to have quantum computing, more advanced AI capabilities. Our predictions, we aim to be 95 to 98% accuracy. So to measure success, measuring success for us is to see that, oh, is there an area of the world that isn't currently endemic or currently being affected by malaria? And our prediction would tell us that, oh, yes, it is. And soon in that future, if that is the truth and we do see this result, and we see this result over multiple regions, multiple areas, and multiple places, multiple communities, and we're able to just save at least one life from it, that is success for us.
Definitely. Thank you so much.
This sounds very similar to the weather modeling systems that are used by meteorologists throughout the world in developing their forecasts. This is a potentially breakthrough technology.
Thank you, Tom, and thank you, Jaylani, for the excellent questioning. We're very appreciative to share our insights with you guys.
Thank you.
I have a question. So I see like the way you guys are able to predict the outbreaks based on the climate, I think is a great idea because, as you mentioned, the correlation with the humidity and stuff. But my question is, the people living in these regions often live with the expectation of encountering malaria, but the issue is that they can't gain access to treatments and preventative care. So how would this innovation be able to address these disparities?
You have to partner with some of the other people in this room.
I apologize. Could you repeat your question? Just the last bit, it was a little unclear to me.
Sorry, I had said, how would the data and warnings about the outbreaks be able to address the disparities of treatments and preventative care for the people expecting to encounter malaria?
Yeah, of course, I can answer that. And I'd love, once I finish this this short answer, Aldo, who actually was looking a lot very into how can we disperse these materials, can touch up on this. But so in these areas that are going to be low on resources, again, we want to reaffirm that allocating our resources effectively is a goal as well. So that means if we know that an area that's already being affected is under the pressure of malaria, we know that resources need to be allocated there. Then if our prediction says, okay, a community maybe 5 to 50 miles out is going to experience soon immense pressure as well from malaria, we will begin taking preventative measures and allocating resources there as well. And Aldo could actually touch a little bit more on that.
Yeah, so essentially, in terms of information, our predictions will be able to predict where a certain area is about to become an endemic area. And depending on rain patterns, humidity, and other certain forms of precipitation, bodies of water, specifically stagnant water, can, of course, become breeding grounds for these mosquitoes. And essentially, with this information, the people in the communities can basically prepare, or if they see, for example, in the sink, oh, it's filled with water, can they do? Drain it. That way they can avoid these breeding grounds from kind of forming, and not just the sink, of course, but anywhere else around that they see stagnant water that can potentially become a breeding ground, and that will help prevent the spread of malaria.
And I would like to add one point as well, I think is a very key point as well. Aldo pretty much mentioned the base of it, but it's the role of the local governments that we're going to be informing as well. When you inform the local governments about malaria that's incoming to their area, it turns the fear to awareness. And these local governments can actually implement their own public health campaigns, provide funding for those interventions, and enforce policies to better prepare for those upcoming outbreaks. And we believe, again, a lot of areas have different situations and they're going to have their own unique resolutions.
Okay, thank you.
You're welcome.
All right, any other questions? No, well, great job. And then we'll move on to County College of Morris, malaria.
Perfect. Makuta will be sharing her screen for the presentation.
Hello.
All right, you should be able to share now.
Sorry. Do you see the screen?
No, we see black.
Okay. Oh,
Now
oh,
we see your screen. Now we see your screen.
sorry.
Okay, perfect. All right. Okay. Hi, everyone. Thank you for having us today. My name is Elif.
I'm Gio.
I'm Kuta.
We are Team Malaria, and we present to you Sentinel-M, your malaria watchdog that will detect, protect, and defeat. Sentinel-M is an innovative low-cost sensor designed to detect malaria early, helping to prevent its spread. Using simple sensor technology, it identifies a rough estimate of a mosquito's density in the environment with fast and reliable detection. Ideal for communities with limited healthcare access, such as sub-Saharan Africa. Sentinel-M offers a simple, affordable solution to track and manage malaria outbreaks before they escalate. Much like exposure notification systems used during the COVID pandemic, this system will use nature's cues to alert healthcare providers, government officials, or local news networks about a potential hotspot so people can act fast, get vaccinated, and protect themselves from infection.
Sentinel-M, the tiny but mighty mosquito detection system, uses small and inexpensive sensors and can be installed inside or outside homes, clinics, community buildings such as schools or mosquito breeding sites. Since female mosquitoes are the ones who spread the infection, we can target them by incorporating a sound sensor because we know that mosquito wing frequency differs by gender. Other smart sensors can detect mosquito motion, CO2 temperature, and humidity. Our smart sensors detect mosquitoes before they strike. Our sensors will continuously collect data and send it to a central hub, such as a mobile app, local server, or a cloud-based platform. Also, with the advancement of artificial intelligence, the data Sentinel-M collects can be processed and used to train machine learning algorithms that can provide accurate predictions of mosquito activity. When our sensors detect increased activity or conditions favorable for mosquito breeding, an alert system will be triggered and a live hotspot map can be generated. Health officials, community members, and you will be notified when mosquito activity spikes. No more itchy bites and a smarter and faster approach to protection. Stay ahead of the bite and let Sentinel-M do the fight.
Imagine smart sensors costing as little as $10 to $50 each, detecting mosquitoes in real time. These sensors use affordable materials and require minimal energy, making them ideal for large-scale deployment. By leveraging mobile networks such as apps, SMS, or offline cloud storage, communities can receive instant alerts, with data sent to local hubs like hospitals or health centers. This ensures rapid response and targeted intervention, even in areas with limited connectivity. A small pilot of 100 sensors can be launched for just $20,000, covering hardware, data infrastructure, and training. Adding over 1,000 sensors could cost about 100 grand per year. Most importantly, this solution empowers local communities due to affordable microcontrollers like Arduino, along with solar or battery packs. Even small households can set up their own monitoring systems for as little as $25.
You are muted.
Oh my God, I'm so sorry. To bring this to life, we simulated our product using Arduino software. This simulation includes a motion sensor, and as you can see, if a mosquito were to fly nearby, the lights would get brighter and the Arduino would send a message back to the central hub. Another way this can work is that the light would stay on to attract the mosquitoes, and the motion sensor could estimate the mosquito density in the area. Next slide, please. In the bottom, you can, I think, switch to the next slide, or you can use your arrows. Yeah, like that there. Okay, please play the video. Okay, when we said it's customizable, we meant it. We can add a variety of sensors ranging from sound to temperature, as this simulation shows. If temperatures were to increase beyond the threshold, which we can set according to the location it's installed, the lights would get brighter to attract the active mosquitoes, because we know mosquitoes like warm weather. And it would send a message back to the hub to let everyone know. In the end, it should give us depictions that look like the image on the bottom right, which means that it can detect malaria hotspots in real time. With our accessible tool and a network of first aiders responding to alerts, we can protect, detect, and defeat. Because if we stay ahead of the bite, we stay ahead of the fight. I'm sorry. With SANO-M, the solution is simple, and we say malaria can buzz out of here. Thank you.
Thank you.
Great job. Did you have more slides? Because we only saw really 2 and then that video.
Yeah, no, that was it.
Okay,
The 2 videos were, yeah, okay.
Okay, all right. So what would you, I guess, okay, you have how many of these would you need?
So they can be put like outside buildings and stuff. So you can put it where you need it. Because some areas might need it more than others. And at the same time, if there's like one and there's like lights on that can attract mosquitoes, then you don't really need that much. So like you can say a mile radius, 2-mile radius, something like that.
Okay. Tom, questions?
So have you given more thought to the various environmental factors that mosquitoes are naturally attracted to besides the lights?
Well, yes, if there were houses or communities that are next to the water, then there's more likely to be more mosquitoes there. So that means this device would detect them. But other than that, no, it's like a natural occurring thing, and we just put the sensor and it would sense like the mosquitoes and it would have lights on. Like those bug traps that have lights on. This is the same concept, except that it's just a detector. And then it just lets people know that, oh, there's like a higher activity of mosquitoes, stay out of that area, or guard yourself by getting vaccinated or stuff, or using insecticides.
So if there are not a lot of mosquitoes being attracted, you're assuming the density is low. So
yes, exactly.
It's a safer area. Okay.
Exactly.
That's viable.
All right, anyone else? Questions?
I was just wondering, when it came to the mosquitoes being attracted, what would, like, customizable is great, but is there no reason to just have every sensor and have the best check the best way to check for the density of them?
Oh, okay. So when we say customizable, we mean like there's so many sensors that we can use to detect the mosquitoes. There's a motion sensor, there's a temperature sensor, so that if it can be used with the motion sensor. Honestly, these Arduino like devices, they're like you can take things out, put things back in, like they're so easy to use. So you can also add sound sensors, because we know like female mosquitoes are the bigger problem. And we know that their wing beating frequency is different from male mosquitoes, so they sound different. So we can particularly target female mosquitoes in an area by adding that sound sensor, and they all cost around the same. Like they're really cheap. So like this keyboard is like really, there's humidity sensors, there's carbon dioxide sensors, because you know that all these things, mosquitoes are attracted to them. So if they're higher in some areas, they're more likely to be there.
Okay, I see. Thank you.
Does that answer your question? Okay.
Anybody else? I love that you can target the females.
Yeah, that's interesting that you have that information available. I didn't know there was a difference in the way they beat their wings.
Yeah, it's their wing beat. I think it's because they're sexually dimorphic. Females are normally larger.
Very interesting. You've done your homework.
Yeah.
Yes.
Anybody else have questions? Okay, then I think. Thank you so much, you guys. That was a great presentation. Our next team is from William Patterson, the Trespassers. Francisco, Samantha, are you guys here?
Yes, we are. Can you make Samantha the host? She will be the one sharing the presentation.
Sure.
Okay, I'll be sharing my PowerPoint.
All right, you should be able to share now.
All right. Can we see that?
Yep.
Yeah.
All right.
Well, good morning, everyone. We are Samantha Fernandez, Francisco Perez, and Roma Parvin, and we're looking for innovations for malaria. We're specifically looking at the RNAi treatment. Malaria is a problem all around the globe. Next slide. And even though that we are here in the US and we are not affected by malaria, it is our duty and job as scientists to still innovate for this problem. We do know that malaria is not caused by the mosquito, it's caused by the plasmodium that is that's gonna be carrying the mosquito. And once a mosquito bites a person, then the parasite is gonna move to the human liver cells, where it's gonna be living there until it's ready to close out of there. Once it's mature enough, it will enter our bloodstream and go into the red blood cells and form a vacuole inside them and use the PTX protein to release responses. So therefore, we were looking at foreign to targets to either use looking at the human liver or the PTX proteins, and we focus on the liver.
So today we're going to be focusing on the liver malaria. So the mechanism goes as follows. Once a female mosquito bites its host, the plasmodium parasite is released in the bloodstream. After a few hours, the sporozoites will invade the hepatocells in the liver, which are responsible for metabolism, detoxification, bioproduction, storage, and immune function. There will be a 7-day grace period following this for the parasite to develop and replicate, producing for each protozoite about 10,000 blood-infected daughter parasites. These daughter parasites rupture the hepatocytes to then be released into the bloodstream. That will begin the asexual replication in the circulating red blood cells, which is when clinical symptoms begin to show. During this stage, the parasite is at its most vulnerable because of the number of sporozoites in the body, which is what the therapy will be targeting.
RNA interference treatment for malaria involves using RNA interference technology to target and silence specific gene in malaria parasites. How will it work? By using RNA interference to block proteins CSP and TRAP that helps the parasite to enter liver cell, preventing infection from starting. Also using it to target UIS3 and UIS4 gene, which the parasite needs to build a protective bubble inside the cell livers. Without it, the parasite dies. Also, we're going to be focusing on shutting down gene FAS2 gene that helps the parasite get nutrition, stopping its growth, and preventing the parasites from making new infection form, which focus on gene LS1 and SER gene, stopping it from moving it from the liver to the bloodstream, where malaria symptoms start. Why we thought this would be a better option, as it prevents infection before the symptoms arrive, unlike current drug that treats after malaria already spread. Also, it has lower resistance risk because it can target multiple genes, while parasite have developed resistance to most traditional drug. Also, it has fewer side effects as it doesn't interfere with human cell function, and its long-lasting protection unlike current treatment that requires multiple doses. Next slide. Next slide. Okay. Initially, development of RNA interference-based malaria treatment is costly, but collaborating with global funding and existing malaria programs, and as technology got advanced, it could lower the expense, making RNA interference therapy more accessible to the future. As we already know, many lives has been lost. So by focusing on this, we could help other people and lower the death rate. Overall, RNA interference theory for malaria could be a game-changing approach by stopping the parasite before symptoms even begin. And its precise, long-lasting effect and lower resistant risks makes it a powerful alternative to traditional drugs. Next slide. Thank you.
Great job. Congratulations. That was a really, really good presentation. So let me understand this. You're targeting 2 proteins and 5 different genes?
Yes, we're going to be focusing on most on blocking the CSP and trap that helps the parasites to enter the liver cell. The liver is where it gets developed. So if we stop it at the liver, then it wouldn't go to the bloodstream because it's going to be harder to stop it if it goes in the bloodstream.
Okay.
And similarly, we know that the mosquito is not the one carrying. Therefore, if you are able to prevent mosquitoes from acquiring the plasmodium, that means that less infection is going to be passing around.
Right. That makes sense. All right. And these are genes then that are being targeted that don't impact any other functioning of the liver?
No, it's just gonna be functioning because like we're gonna be using because like RNA is like you make the same copy of the genes, but then you work it and like alter it so that it doesn't affect the body, like it doesn't do its function as it's supposed to.
And as well, the plasmodium is feeding on the lipids of the liver cell. Therefore, if we target that, they're gonna die of starvation, while our liver doesn't have their functions ruined, or it doesn't affect us.
All right. Tom, any questions?
So is this RNA interference therapy, does this already exist?
No, it's something good they're looking into because RNA treatment interference they haven't worked on it. They were mostly focusing on cancers now, but it's not something they have started yet.
Interesting. Way to go about this.
Yes, it's very innovative. I like that.
Thank you. Anybody else has a question?
Anybody? Questions? No? Okay. Well then, thank you. Trespassers, up next, we have from Montclair State, Buzz Killers. Genesis and Yuma. Anybody?
We're here.
Okay. Are you going to be the one sharing, Genesis?
No, I think it's Helen. Let me ask.
Anybody can shout out who's going to share, and then I can just make them the co-host.
I will be sharing.
Okay. So I will make you co-host. So you should be able to share now.
Okay, 1 second. Can you guys see?
Yes.
Oh, okay. Okay, so this is our aquacore. It's a pump system, and that we have come up with for the systemic presentation for this year. Okay, so some of our key goals when going into this were to create a new and innovative approach to malaria that would work in the long run, so we wouldn't have to continue to revisit it every time, because that's what we've seen happen with the vaccines. And we would also like to stop the production of the malaria carrying mosquito, as we felt that by stopping the reproduction would also stop them from carrying it to humans in these affected areas.
Okay, so our innovative idea is the following. Taking control of the fight against malaria, we introduce a solar power water pump that serves as a still water disruptor that will not allow the reproduction of the Anopheles mosquito and therefore reduce the rates of malaria cases in the region. AquaCore is eco-friendly, easy built, and long-lasting. We are committed to protecting vulnerable communities by delivering an innovative and sustainable solution that eliminates reliance on temporary interventions. We're not just fighting a disease, we're fighting for a world where no child's future is stolen by malaria, where families live without fear, and where communities can thrive without the constant threat of illness.
This is the model of the prototype where it has a solar panel, a suction motor pump, the lithium battery, the tubes, tube 1 and 2, the mesh, and an MPPT charger control. First, we would identify areas with high cases of malaria. One that I researched was Nigeria. Second, we will introduce the pump system, and then we will compare the number of cases of malaria before and after the system was introduced. This is an easy build, and it will be shipped in.
Helen, we lost your audio.
Okay, so now I'm gonna be talking about a really crucial part of the aqua pump, aqua core pump, which is the materials. One of the main goals when we were talking about the manufacturing process of the aqua core pump is we wanted it to not only be affordable, but we wanted it to be something that would last for a really long time, especially in a very dry climate. Here in detail, you can see the very different materials that would be used for every step of the manufacturing process of the aqua core pump. The main source of energy for the pump is solar powered, and we would do that by using solar panels. And this is how we would ensure the supply and the longevity of clean water for the communities. There are a lot of other additional materials like battery screws, the plastic tubing, and the stainless steel panels, but these are extremely important to the pump and protected from damage and harsh conditions. The total price of making one pump would be $510, and even though it does look a little expensive, the positive effect it will have on communities in the years to come will surpass the price of the struggle of medication and the medical care on these disadvantaged communities.
So, in order to distribute this, we would do a sort of easy assembly kind of packaging where we would send them disassembled, to these areas, and then have step-by-step instructions on how to assemble them that would that would be visually easy for people to understand, to cut down on costs on having to have someone else go and build it. We would only send the essential components and collaborate with local businesses for assembly, just in case, for some reason, it could only be sent to that business. We would send it to them, and then they would further distribute to the people. And then we can also partner with humanitarian organizations, such as Project Hope, UNICEF, and the Red Cross, Red Crescent, to include our pumps in their already established shipments to these areas.
For funding, our team is looking forward to partnering with the Bill and Melinda Gates Foundation as well as USAID for funding and distribution of AquaCore, since they both focus on funding preventative innovations like our pump system. The Gates Foundation specifically has a goal to completely eradicate malaria by 2040 with new and existing preventative innovations, which is relatively close. And by pitching our system to the foundation, we hope to be of assistance to that goal. And in partnering with USAID, we hope to distribute AquaCore in the 16 countries in Africa that they are already present in, since they already have a focus on financing and supporting new and preventative innovations. We would like to pitch AquaCore to USAID as a staple to eradicating malaria. Thank you so much.
Great job. So I just have a question. Is this something that you would be thinking of a per household, like each household would have a pump?
We were thinking that this would be something that you would put, because we already spoke about how the mosquitoes reproduce in still water and in large bodies, also in the houses. But we were thinking we would put this where there would be large bodies of water, because that way the pump isn't necessarily in the middle of the thing, so it doesn't in the pond or whatever, so that it doesn't affect the ecosystem of that certain pond, but it would instead be on the side, sprinkling water over the pond to disturb the water, and that way, and it would, as you can see in our prototype drawing, it uses sunlight for energy, so that way you don't have to worry about connecting it to anything, and the pump would work on its own, and you just set it up and kind of forget about it kind of thing. That would work in larger bodies.
Okay, so you're basically taking water from the pond or pool or puddle or whatever, and then spraying part of it back over it?
Yeah, just to distribute, just to disturb the water.
To disturb it. Okay, very interesting. I didn't understand that initially. Okay. And is there a minimum size? Because that seems like a pretty massive pump.
We were just thinking of this as the basic size. It could be made smaller or bigger, but this was just the standard that we came up with initially.
Okay, excellent. Tom?
Curious, do you happen to know what the normal ecological function is of this mosquito in the areas where it is most endemic? Because you're liable to encounter some serious resistance from a number of different agencies if you don't know what the normal function is of this mosquito before you go killing it off.
At first, when we were designing the idea of this prototype, we did understand that there will be minimum side effects to the mosquitoes because we will be killing them, but we did further research on how these mosquitoes help the ecosystem. We just focus on how they're transmitting malaria to people.
Okay. Thank you.
Anybody else have questions?
Hi, I have a question. So The reason why my team went with a more computerized technological approach is because when specifically dealing with mosquitoes, not the malaria variant or parasite, it's very difficult to predict what environmental factors can influence this change or when you start manipulating ecosystems and these small body populations, you have no idea what could happen in regards to the ecosystem as a whole, such as the predator and prey dynamics, the disease transmission. So I was just wondering if you guys had a plan for that, just in case, let's say, uh, affecting the mosquito populations in a large region could have scale effects further down the line, because you said this is a more long-term perspective.
So that's why we have brought up the idea of doing a sort of trial to see how that would affect the ecosystem initially, or to see how the pump would work in different areas. But also, when we initially came up with the idea, we looked up like what kinds of effects do mosquitoes have on ecosystems, and they seem to be very minimal. Maybe not here, but more towards tropical areas, because they do, like, we looked up, they like transfer like pollen or something, and bees are there, and also they're there for feeding, but also there's other insects that those animals could feed off of too. Like, that it's not just mosquitoes that they feed off of, so it wouldn't be that big of an impact as we would think. But that is why we had the idea of having like a trial where we would see how things would go down.
I love the idea of a trial, but I would say on that point that you made that uh, not as much of an impact as we think. There's a lot of cases in history that we have added something to an ecosystem or took something away, and it was either in a small area and it made a much larger impact than they thought.
All right, any other questions? Nope, okay, well, thank you. Great job. I like your innovation. I like when we have we've had a lot of very, very different types of approaches, which is fabulous. Thank you. And I think we have a few teams that haven't shown up, but next is the William Patterson Malaria Menaces. Are you guys here?
Malaria what?
Menaces.
Menaces.
Menaces. Sebastian, Princess, are you guys?
Yeah, we're here. Could I be the one to share our screen?
Of course. You should be able to do that now.
Okay. Okay. Can you guys see my screen?
Yes.
Oh no, the screen. Hang on, I gotta move out of the way. Okay. So hi, my name is Jeanette. Guys, are you there?
Hi, my name is Sebastian Norris.
Hi, my name is Ileana Abreu.
Princess, are you there?
She doesn't seem to be in the room anymore.
Oh, she must have gotten booted out. Okay. By any chance, could we go next just to make sure our team member can get back?
You guys are last at this point.
Oh, okay. This isn't counting towards our 5 minutes, is it?
Yeah.
I can read her parts.
I'll
Okay.
stop until she comes back.
Okay.
Yeah, Tom, if you could restart, that would be great.
Well, restart the whole thing.
And
Okay,
if you
thank
guys
you.
could
Yeah, we
turn
haven't
on
done
your
anything
camera.
yet, so that's fine.
Yeah,
If you
I can.
guys
Oh, I'm
could-
sorry. Let me turn on my camera.
All right, so you can start whenever you're ready.
Okay, hi. So, Okay, so our project is on the genetic modification of Amyl anopheles mosquitoes to reduce the contraction of malaria in humans.
Okay, malaria mostly impacts people living in poor rural areas of Africa. In 2023, 95% of all malaria deaths occurring in the WHO African region. Children under 5 and people experiencing malnutrition are most at risk. While there are many innovations that help prevent contraction of the disease, they are often inaccessible to the people who most need them due to the issue with distribution and the inability to afford them. The use of bed nets is higher in better off population in poor households, and poor households spend significantly less on all forms of malaria preventions compared to their richer counterparts, including bed nets and insecticides.
While there are 5 Plasmodium parasites that can cause malaria, Plasmodium vivax and Plasmodium falciparum are the most prevalent. Plasmodium vivax accounts for the most malaria cases worldwide. However, Plasmodium falciparum causes over 90% of malaria-associated deaths. Therefore, our project will be focusing on combating P. falciparum. Additionally, all malaria-causing Plasmodium must infect mosquito vectors in order to breed, with the female Anopheles mosquito being the most prevalent vector.
This slide provides a visual of the Plasmodium life cycle. We will be targeting the gametocyte phase, as this is the stage that is present and engages in breeding within the mosquito midgut.
Plasmodium falciparum has a surface protein called PFS16 that reduces the activity of midgut caspases, essential in the mosquito kinase immune pathway. PFS16, therefore, is an important immune evasion structure in the parasite that allows it to invade the mosquito midgut to breed by actively silencing the mosquito's innate immunity and its interactions with the midgut epithelial cells. We propose genetically modifying the TEP1 protein, which is a naturally occurring complement-like protein in the mosquito immune system, to target and bind to this PFS16 protein. This will not only sabotage the parasite's immune evasion strategy, but also interfere with its ability to infiltrate the midgut, which is essential for the Plasmodium to be able to breed.
We will begin the modification process by using X-ray crystallography to study the structure of the TEP1 protein and identify possible alternate binding structures that will increase its affinity for PFS16.
We would then generate mutant primers designed to introduce mutations in the binding domain coding region of the TEP1 gene. This would be done to perform site-directed mutagenesis, and a library of TEP1 variants can be created to increase the chances of finding a TEP1 protein that targets and binds to its PFS16.
We could test the affinity of our mutated TEP1 via immunoprecipitation, in which proteins with the ability to bind to PFS16 can be separated and purified. Western blot tests for the proper expression of the protein.
CRISPR-Cas9 can use a designed gRNA to target and knock out native TEP1 genes through non-homologous end joining. Then the system can be used to knock in the mutated TEP1 gene. We can validate the insertion and gene expression using collected cell samples and techniques such as conventional PCR followed by gel electrophoresis, Sanger sequencing, and as previously mentioned, Western blotting.
Said these gene editing technologies are very expensive and not accessible to everyone. However, there are funding in the form of global grants to help pay for this research. TER covers laboratory and field testing of mosquito genome editing techniques, facilitates the expansion of successful interventions in regions heavily affected by malaria, and fund evaluations to measure the impact on malaria transmission and public health outcomes. WHO and the Global Fund to Fight AIDS, Tuberculosis, and Malaria work together with research institutions and finance programs essential for achieving malaria elimination goals through innovative tools and strategies. This program also strengthens health systems and research capacity in regions with high malaria prevalence. Our GMO strategy aims to be environmentally friendly and minimize ecological disruption by ensuring the mosquito itself remains unharmed. Additionally, this modification helps fight the burden of the parasite of the mosquito, which should be favored by natural selection. The ability of the mosquitoes to distribute themselves helps fight disparities of access to treatments and preventive materials, does not place financial burden on the families to provide their own malaria prevention. And since these changes are designed to be passed on to future generations, we can ensure long-term malaria prevention without multiple interventions.
We would like to thank Dr. Eliana Antonio, the GS-LSAMP coordinator, William Patterson, Dr. Kendall Martin, our faculty advisor, and the GS-LSAMP community for making this presentation possible.
Thank you so much. That was great. All right, so I'm very impressed. You said specifically that Plasmodium falciparum has PFS 16. Do you know if that's a universal membrane protein, or is it just that particular strain of mosquito, species of mosquito?
That's a good question. That protein was taken from a NIH source that was just published a little less than a year ago. So the structure is still being studied. Its primary focus has been on Plasmodium falciparum because of the fact that that specific Plasmodium is responsible for over 90% of fatalities. So in our research, we would be targeting that specific Plasmodium species with this protein because in the moment it's only been identified on their surface as the research is still ongoing.
Interesting, very interesting. Okay, Tom?
No, so it is interesting. I have no questions.
All right, anybody else? That was highly technical, but I hope you guys have some questions.
I have a question. How would you know that your gene editing is working?
Yeah, so we can make sure that there's proper insertion and also that we're cutting out the right gene by using, as mentioned in the slides, conventional PCR followed by gel electrophoresis. So that, first of all, make sure that the inserted gene is the actual targeted site of TEP1, the native TEP1. And then we can also use Sanger sequencing, which would confirm the nucleotide sequence and fidelity of the insertion. So we have chromatograms and we'll get a file from the company that does the Sanger sequencing, and we can just compare and see if the nucleotides are there. And then also Western blotting, which also would confirm the presence of TEP1 proteins actually folding and being made properly.
Yeah, because you're talking about designing something for a protein that hasn't even been determined how it folds, nothing about it, right? It's a pretty newly discovered protein.
So at the beginning, we would perform structural analysis. So that's what the X-ray crystallography would be for. We would start looking at the structure of it first, see any binding domains and what can be manipulated to make it bind or have a higher affinity for PFS16. Because since it's a surface level protein, we think it can bind there.
And just to add on to what Sebastian had said, there is a study that was published on the PFS16 protein where they have studied how that protein works in the immune evasion and how it's interacting with the cast spaces of the mosquito to infiltrate the epithelial cells. So there is some understanding of the protein's function, but we designed our experimental protocol under the assumption that there are still unknowns.
Yeah, really interesting. Very, very interesting. Anybody else have questions? All right, you guys did a great job. I think as most of you know, well, first of all, happy Pi Day, everybody. I forgot it was
Happy Pi Day.
Happy Pi Day.
So as most of you know, we've GS-LSAMP is sort of under target from the current administration, since we definitely covered DEI. And so GS-LSAMP has yet to be refunded. We are currently in a renewal process and have not yet heard whether or not we'll get any additional funds moving forward. So we are out of money, GS-LSAMP. So we are going to provide stipends. Unfortunately, it's only to the top 3 teams because we're not paying for the stipends. The chancellor of Rutgers Newark has very graciously agreed to provide stipends to the top 3 teams. So the top team will get $100, the second place each. The second place team will get $75 and the third place team will get $50 per. And I know this is less than we've given you guys in the past, and I'm sorry. If we had our own funding, we would definitely be providing you with more, because I think you guys work really hard at this. And show of hands, how many of you thought this was a great experience?
It definitely was. Definitely.
Did you guys learn things that you didn't expect to learn? And so if you could say one thing about this experience, what would it be? Anybody can just yell out.
I think it would say it's thinking out of the box, because trying to also have in consideration like the population, where they were located at, that was amazing. That was like thinking out of the box and putting everything that we learned in the classroom into the like real life, even though we didn't get to make it in person, but yeah, it was that.
Excellent. Anybody else?
I will also say the experience served to learn about what's out there in the future. Like this mainly applied to my group, but I was able to visit Drexel University, and I was talking to the faculty that has 3 different labs on malaria there. It mentions that even though that this is not here in the US, like they still, as scientists, is the duty to do the innovative research. So it serves also for the drive to always seeking hunger for science.
I believe it's so wonderful, this opportunity that we are challenging ourselves, and we realize that we can do more than we can think. And that is a more beautiful thing about all this job. And thank you for this opportunity.
That's great. Anybody else want to say anything about this experience?
I would say on the behalf of Montclair State University's team at Astro Team, that we were given a really tough challenge. And, looking at it from all perspectives, it looks like what can like a couple of undergraduate students do, right? But, we really put our heads together and we just try to think of it as outwardly as possible, like someone mentioned before, out of the box. And it really tests the strength of the will and the way that the students can work together.
So teamwork is one of the most important things, both in this challenge and in science. Like, you can't do science in your own little isolated box. You have to work with other people. It's imperative. Otherwise, we reinvent the wheel every single time. So that was one thing that's really important for you guys to get from this experience. I think the other thing that I really, really hope you got from this is that you have more knowledge than you think. The things that you learn in the classroom, you can actually apply to real world situations. And I also want you to know that science is creative. And I think that as undergraduates, you don't have the opportunity to see that very often, because you're in classrooms and you're doing prescribed experiments and you don't get to think up things. But science is creative. And if it's not, then you're doing it wrong, right? So all of these things, I hope you continue, and I hope this impacts the way that you do science, do STEM going forward. All right, so I am going to go into a breakout room with Tom Van Aken from the B2B, and we're going to make our decisions about the winning teams. So take like a 10-minute break and then come back and we'll announce the winners, okay? All right. So.
Okay, so
I don't know how to go into a room.
Join just popped up. So have you?
Yes, so you should join.
Okay.
All right. So we did our little debate. You guys all did a great job. I really want to stress that. Amazing job. It is always incredible to us how varied the ideas are and how well and thought out they are. It's just incredible. And I encourage all of you to keep innovating. And for some of you, take these ideas, see if you can run with them, right? Because they're great starting points. So we're going to start with the third place team. In third place, from Montclair State University, we have Ad Astra. You can do your little clappy symbol, right? Raise your hand. See that? Excellent. Thank you. I see one. I don't know how to get back there. Oh, here, react. You should be able to react. Yay.
Thank you.
Great job.
Thank you, guys.
Great job.
Thank you so much.
Thank you.
All right. In number 2, in the second place, we have the Rutgers New Brunswick team. Are you stemming? Matthew presented. Great job. That was Project Ripple with a water agitator. I don't know if he's still here. I think he had a class. But again, great job. You guys all did great jobs. And then in first place, we have from Passaic County Community College, Team CCCC, Judith, Amara, and Brigitte. You guys did a great job as well. So thank you all. For those of you who are in the top
so much.
For those of you in the top 3, I'll email you with information that I need in order to get you your stipends, okay? Thank you all for participating. This has been, we had the most teams this year than we've ever had. We were supposed to have 15, but we ended up having 12. 3 teams didn't show up today. You guys did a great job. I'm always so amazed and excited to see what you do. So let's see, I'm trying to take a screenshot, which never works exactly as I planned. So let's go here. Let's do that. And let me, before anybody leaves, let me get another shot.
This was really well done, guys. I just want you to know that I'm in the process of writing an annual report, and this is going to be one of the major accomplishments for this entire year. And I'm going to hide. And Chris, I want to tell you, without you, we would never have had this thing. So hats off to Dr. San Miguel for having thought to develop this wonderful program. Show your STEM innovation. Without her, this would never have happened.
Thank you very much. Yeah, I think it's a great one. I'm really pleased. Can you guys all turn on your cameras so I can get a screenshot of everybody?
Let me do the same. Let me get a picture.
I'm so proud of all of you guys. You're just amazing. You're going to do amazing things.
Absolutely. And even the questions you asked of other teams could not have been any better phrased.
Okay. Thank you.
Thank you.
Thank you.
Thank you.
Have a great rest of your day. Have a great weekend.
Have a good weekend.
Take
Thank
care.
you so much.
Bye, Judith. You guys are so great.
Chris, thank you.
Of course. This was you know, this is my pet.
Congratulations, Judy and team.
This is Linda.
You, Linda.
Thank you. Great presentation.
Nice job.
Fabulous presentation.
Thank you so much.
You're welcome.
Thank you
so much.
You really are. What a tremendous job.
Thank
you.
Have a good day. We love you. Thank you.
We love you.
Bye.